Hello, everybody. This is Karin Abitorabi from Novartis. I work in cell and gene therapy, and I'm very happy to introduce you to the new Therapeutic Modalities session. We will start this session with Dr. Ajay Kopade, and he will tell us a little bit about the development of Teclantis and other novel derived formulations. Ajay?
So I'll be presenting a novel nanoparticle platform technology for the passive targeting of anti-cancer drug paclitaxel. The product is named as Teclantis, and the platform technology is called as nanotecton. So before I start my presentation, I have a disclosure to make. The disclosure is that I am a full-time employee of Pharma, and the views expressed in this presentation are mine and do not in any way reflect company's views or strategies about the product. With that, I'll start the case study on the development of Teclantis, and we name it as a small is a new big. And if time permits, I would also go to the other novel derived formulation in the next 15 minutes. So let me begin with the story of Taxol. The Taxol is one of the most successful drugs for solid tumors. It ranks first among the anti-cancer drugs. And it is also established standard of care for many of the advanced cancers such as breast, lung, ovary, etc. It became a blockbuster just because it has significantly higher response rates and survival advantages in wide variety of solid tumors. This drug was approved in 2002. And since then, until 2020 today, I mean, this is one of the widely used drug in many kinds of solid tumors, including many of the off-label uses. Today in USA, estimated about 100 to 115,000 patients are treated with paclitaxel per year, despite many of the new modalities, new NCs have come into the market. However, the problem is that there is a limitation in the currently available Taxol therapy. And what is that limitation? Let us understand what is Taxol. The Taxol contains paclitaxel. Paclitaxel is a water-insoluble drug, and it requires some kind of a solubilizer to solubilize paclitaxel. And at that point of time in 2002, cremophore was one of the important ingredients which solved this solubility problem. So the product was solubilized in cremophore. However, for the solubility, it required a very large quantity of cremophore. Now, if I need to give us context about this, then for a single dose of paclitaxel of about, which is 175 milligram per meter square, on an average, 20 grams of cremophore is injected in the body of a patient. Now, what this cremophore does, I mean, cremophore releases histamine, activate complement system, and therefore there is a need of pre-medication with corticosteroids or antihistamines. As it is shown in the figure, the cremophore micelles do not disturb, do not dissolve, and therefore it display a non-linear pharmacokinetics for paclitaxel, and therefore there is unpredictable effect of dose escalation or de-escalation. Because of the very high concentration of the surfactant, the limitation is that you cannot use any kind of plastic infusion pack, need special infusion bags. At the same time, the solubility is not optimal, and therefore there are chances due to the temperature variation, paclitaxel can precipitate out, and therefore it needs an inline filter. So due to the high concentration of cremophore, it also leads to various severe toxicity like neutropenia or peripheral neuropathies. Now, some of the example which is given is illustrated in the pack insert, which is shown on this slide. And all these points are actually clearly mentioned on the pack insert. And in spite of these limitations, actually, it still continues to be a widely accepted anti-cancer drug because of its efficacy in wide variety of solid tumors. Now, how do we solve this problem? This problem was later solved by a product, a reformulated product called Abraxane. And let us go through the Abraxane story. Abraxane was the first reformulated paclitaxel product. It was approved in various countries. And the product is a lyophilized injection, and it is cremophore free, and therefore it solves all the problems which are related to cremophore. It is albumin bound paclitaxel. It does not require pre-medication. It has a short infusion time, a high maximum tolerated dose, and also displays improved efficacy compared to Taxol because of its ability to give higher dose. Now you can see in this figure, it contains paclitaxel coated by albumin. So these are the kind of nanoparticles which are present in the Abraxane product. Now, so if you have solved half of the problems, is there an opportunity to improve the product further? That's the question we asked ourselves. So we went through the pack insert, and on this slide, there is an excerpt from the pack insert of the approved Abraxane label. It shows that the Abraxane uses a biological source of albumin, which may cause severe hypersensitivity reaction, sometimes a fatal outcome. It also contains albumin, which is derived from human blood and therefore has a theoretical risk of viral transmission. Besides this, there is another limitation which we can see is that the preparation for the IV administration is quite complex and time consuming. And this is again an excerpt from the label, which clearly mentions how do you actually reconstitute the lyophilized Abraxane vial. So it has to be slowly injected for a minute along the vial wall, and then it has to sit for 5 minutes, further swirl for 2 minutes, and then if you have a foaming or clumping, then keep it for 15 minutes. And if you suppose the reconstitution is to be done in an aseptic area and it is not done properly, you are wasting the vial. And similarly, 5 such vials are to be made for a single dose, and all these vials has to be withdrawn after reconstitution and filled in an empty bag. So it's such a complex procedure. So what are we doing? Actually, we are solving one of the problems of Taxol, at the same time bringing up another set of problems due to the new improved, so-called improved product. Now, we at Sun Pharma thought about it and thought whether we can solve problems of both the product Taxol as well as Abraxane. What does that mean? That means that it should be easy to use as paclitaxel. It should not have chemophore. At the same time, it should remove the complexity of this admixture procedure of Abraxane. And therefore, we came up with the technology called nanotecton. It's a self-dispersing nanotech platform technology containing, which solves a very trivial problem, which is an insoluble anti-cancer drug using a kind of a building blocks which synergistically form a nanoparticle. We call it as Teclantis, and in short, I may call this product as PICN. This is paclitaxel injection concentrate for nanodispersion. So what is it? So it contains paclitaxel, of course, and a set of GRAS excipients, a fatty acid, that is a lipid 1, and a cholesterol ester, which is as a lipid 2, and a GRAS polymer, which is, in all these ingredients have been used in past for an intravenous injection. Therefore, there are no safety issues. They're all dissolved in a solvent, and you get a clear colorless solution product, which is shown in the panel on the right-hand side, a small vial containing a clear solution. And what you need to do is just withdraw this concentrate from the vial, inject it into an infusion bag, and shake it so that you get nanoparticles which are quite uniform and dispersed like Abraxane. So how this product look like? This product looks like Taxol, which is a pre-concentrate. And upon reconstitution, similar to Taxol, it looks like Abraxane, which is a nanoparticle. So it solves both the problems, and it has properties of both, good properties of both, and which also removes all the drawbacks from both the formulation. So it contains no protein, no chemophore. It is easy to use. It has all the excipients which do not add to the toxicity. It's a quick step procedure, and it also produces a stable nanodispersion. Now, what's the science behind it? What's the physical chemistry? Now, we have done a simulation in a Strodinger simulation software, and we saw that when we mix them together, they actually, all the ingredients come together and form a nanoparticle. So typically, if you have a paclitaxel solution and if you dilute in a dextrose, it would precipitate. Now, in the presence of the lipids which we have chosen and the polymer, it self-assembles. And the interesting part is all these ingredients, if they stand alone mixed with dextrose, do not self-assemble. So they are either water soluble or water insoluble. But when they come together, they have a complementary interaction. That is, the hydrophobic lipids interact with hydrophobic polymer, and they form a kind of an amphipathic structure, due to the interaction, and then they tend to self-assemble. And during that self-assembly, paclitaxel also interact with water-soluble polymer, and they self-assemble to form a micelle or a compound micelle, or an aggregate of a micelle, which is a nanoparticle. So in the simulation, you could say all these material come together. For the clarity, water molecules and the solvents are not shown in the figure, but paclitaxel and the lipids tend to come together and form a nanoparticle, which is quite uniform in size. Now, naturally, the particle is small, which can be seen in a cryotransmission electron microscope as a spherical particle. It is non-crystalline, it is amorphous in a readily bioavailable state, which I'll show in the next slide. It has a mean diameter of about 100 to 150 nanometers with a negative zeta potential, which provides stability to the particle. And the particle has a typical behavior, and the behavior is quite similar to Abraxane. So the particle behavior is that it immediately disintegrates, unlike most of the nanoparticles which are used for, say, long-acting or a prolonged release. But in this case, it immediately disintegrates and facilitate an albumin binding in vivo. So what are we doing? We are actually producing a nanoparticle which is like an Abraxane. Abraxane is a preformed albumin nanoparticle, and what we are doing is we are facilitating the albumin binding in vivo, and which is demonstrated in this figure, where the particle size is on the y-axis. On x-axis is the paclitaxel concentration in plasma. So if you add a particle size of 130 nanometer at a concentration which is relevant to a Cmax concentration after a clinical dosing of 260 milligram per meter square, which is about 25 microgram per ml, then the particle size immediately disintegrates and becomes almost a clear solution. But if you increase the concentration above 100 microgram per ml, then you would not see any disintegration, but that would be equivalent to 1000 milligram per meter square dose, which is far, far away from the actually clinical dose. So that gives us, that shows that the particle which we have produced, the nanotecton technology, is actually a albumin-bound nanoparticle or albumin-bound paclitaxel, which is formed in vivo. So eventually, its behavior is when it is injected in the bloodstream, it disintegrates, and it reaches to the tumor sites via both active and passive means. The particles, the intact particles actually leaks through a leaky vasculature at the tumor site, because the size of the particle is 100 to 150 nanometers. At the same time, all those particles which are disintegrated and bound to albumin are taken up at the tumor site by a GP60 receptor based endocytosis across the endothelial cells of vasculature and reaching the tumor cells at the at the tumor site. Again, the spark or the matricellular proteins actually open up, free up paclitaxel near the tumor site, which then is taken up by tumor and kill the tumor cell. Now, having this proof of concept ready, then we have to prove whether or not the PICN particles is keeping their functionality intact. That means there are two objectives to do a preclinical study. One, that new modality does not lose the efficacy. Either it does not lose the efficacy or safety, or maybe it can improve the safety or efficacy of the drug product. And second objective of the preclinical studies is that what are the studies which are required to eventual filing of the product to a US FDA, or for that matter, any other regulatory authorities. So the two important parts, one is preclinical efficacy studies, which is done in multiple carcinomas xenograft in nude mice. Again, independently validated at CRO, so that you are confident that you are either getting an improved effect or at least getting as equal as effect than the currently marketed formulation. Tissue distribution to see that there is a linearity in the pharmacokinetics. Then there is not much difference or a specific difference in the tissue distribution, so that you can see whether or not any particular tissue is going to have a different kind of a side effect, and special toxicity studies required by US FDA. Now, this on the next slide, I'm showing you a representative graph where we see that in some of the tumor models, you can actually see the PICN is behaving better than the NAB paclitaxel or a protein-bound paclitaxel. It shows almost equal LD10 and LD50, so that the toxicity, the acute toxicity is almost same as that of protein-bound paclitaxel. Following this proof of concept studies, this product went into a phase 1 clinical study, a standard phase 1 clinical trial with the 3+3 escalation protocol in a 3-weekly regimen, and various types of patients having biliary, ovarian, cervical, melanoma, various kinds of solid tumors were treated. Eventually, the mean tolerated dose was found to be 295 milligram per meter square, which was highest among the all marketed paclitaxel formulation. So to give you a context, Taxol has a dose MTD of 175 milligram per meter square. Abraxane has MTD of 260 milligram per meter square. And we have an MTD of 295 milligram per meter square. Following phase 1 clinical study, a small pilot study, which was also enabled Indian approval, was a phase 2, phase 3 study for efficacy, conducted on 180 patients, out of which about 130 patients or 125 patients were evaluable. The objective, the PICN was tolerated both at 260 milligram and 295 milligram per meter dose. So both doses were tested just to see whether we are having an advantage or at an equivalent dose or an improved efficacy at a higher dose. Of course, no hypersensitivity reactions were seen. There were no pre-medication given to the patient. Overall response rate after 6 cycles was quite similar to Abraxane, and even the progression-free survival was also quite similar. Going to the safety part, when the safety was evaluated, the incidence of peripheral neuropathy and a grade 3 or 4 neutropenia and leukopenia was lower for a PICN 260 versus 260, but it was comparable at 295 and 260 with Abraxane. So what it tells us that 260 at an equivalent dose, PICN may have improved safety compared to Abraxane. So following this, the challenge was at this concentration, at this stage, what do we do? I mean, do we go to a full-fledged pivotal clinical trial, and in what indication, or whether we should do a bioequivalence trial, because we saw that both safety and efficacy is more closer or similar to Abraxane, and therefore we decided to go ahead with the shorter route of a bioequivalence trial to a NAB paclitaxel. So a pivotal bioequivalence study was conducted, and about 142 patients were randomized, from which we got 99 evaluable patients. None of the no hypersensitivity reactions were observed. None of the patients were treated with pre-medications. The trials was positive equivalence criteria as per the regulatory requirements for both total and free paclitaxel. So with that, the story of the development of PICN ends. And not last but not least, when you have a technology, it has to be protected by intellectual property, and there are 2 granted patents on the technology, which are all published with an expiry of 2029. So the product is already marketed in India since 2014 and is filed in US and awaiting approval. So the take-home message is a differentiation through drug delivery technology. A small beginnings can have a potential of big outcomes. That's how we close. And I'm not sure how much time do we have, but taking this forward, on the similar lines, there are 2 more technologies which were developed and marketed in US. I wouldn't go into the detail because it would require another case study to be discussed. But in short, just to add on to the repurposing and to the topic, the other product was a glaucoma product. A latanoprost reformulated as a BKC-free formulation in swollen mycel microemulsion technology format. We call it as Lipicell technology, and it is important for use in a dry eye patient. It also endured phase 3 clinical trial in US in about 500 patients, and eventually got approval in US and marketed in US as Zelpros as a first VAK-free prostaglandin. The another product is a nano-micellar product of cyclosporin, which is a preservative-free formulation, a higher concentration cyclosporin formulation made with N-cell technology. It's a non-ionic micellar technology. And the benefit which we obtain was due to the higher concentration, early efficacy. Dry eye endpoints are usually long-term. 6 months to 12 months are required to achieve a significant improvement in dry eye. But in this case, we have achieved significant improvement at the 3rd month interval. This product is also commercialized in US as Sequa. So with this, I conclude my talk, and thank you all for patient listening. Thank you. I hope I have maintained the time, or I'm early, I'm not sure.
A little bit over. Thank you, AJ, for this inspiring talk. Unfortunately, we won't be able to take any questions at this time because now we have Dr. Patrick Baumhof waiting for giving his talk, and he's coming from CureVac, and we're all very interested in hearing what he has to tell us about the mRNAs. Thank you, Patrick. Please go ahead.
Welcome, everybody. It's a great pleasure to speak here at the CleanM. The title of my talk is RNA antibody as a therapeutic option. Since CureVac is publicly listed, I have to show you the forward-looking statements. I am sure every one of you know what's standing there. Therefore, let's jump into the science. We at CureVac are using a revolutionary basic principle. We use messenger RNA as a data carrier for healthy messages. And the idea behind this is that all the proteins that are used in your body are stored as a code, as information on the DNA safely in the nucleus of the cells. And usually, if you want to produce a protein, the information has to be transferred from the nucleus to the ribosomes and translated into proteins. And that is done by messenger RNA. And what we are using now is messenger RNA from outside, a messenger RNA that's produced in our facilities. And we transfer information directly in Situ into the body and produce virtually every protein that we want to today. And in order to use messenger RNA in a therapeutic relevant setting, you need a very good sequence. If you usually start out with just genetic information out of a database, put this into a standard messenger RNA, and use a kit to synthesize and purify the messenger RNA, you usually end up with a messenger RNA that's not very good, that has a very transient and also a very low overall expression. But we at CureVac were working at the messenger RNA sequence for over 20 years now, and we principally mastered these things. And if you sequence engineer in messenger RNA, you can render the pharmacokinetic of a messenger RNA expression inside of cells to a completely different order of magnitude. You can see that we increased compared to the standard RNA, the expression levels by 4 orders of magnitude, and also the pharmacokinetic was dramatically changed. And with such a powerful messenger RNA sequence, you really can think about using messenger RNA for therapeutic applications. In principle, there is unlimited RNA opportunity. You could use this for infectious diseases. I think since COVID, there is quite a focus on messenger RNA-based vaccines. Everyone is looking now into this. In this case, you express the viral antigen by messenger RNA and train the immune system to recognize these. You could also use this for cancer vaccination. In this case, you would use self-antigens, tumor antigens to break immune tolerance and get an immune response against the cancer antigens. And or for protein replacement. In this case, you do not want to induce an immune response against the protein that you express, but you want to use the protein that you express for a therapeutic effect. And one of these proteins that you could express is, for example, antibodies, and that I will concentrate today on. For messenger RNA encoded antibodies, we would use the body as a bioreactor for antibody production. And you would start with the identification of your target molecule that you want to target with your RNAntibody. It could be a protein, it could be a toxin, it could be some tumor cells, bacterial pathogens, or viral pathogens. And once you have then identified your antibody sequence that is principally binding the target, you transfer this in nucleic acid code, produce the messenger RNA, transfer the messenger RNA into cells, and within cells, the ribosomes will produce your antibody. And here you are not limited like recombinant proteins to some aspects. You could produce intrabodies, so intracellular antibodies. You could use secreted SCFVs, secreted antibodies, even membrane-bound CARs, for example. And all of this is just as easy as switching the nucleic acid sequence a little bit. We usually use the liver as a bioreactor for producing the antibody inside of animals, for example. And liver is an interesting target for several reasons. It's a tolerogenic organ. It's the largest organ, and it's well vascularized, so it's highly accessible. And that is quite important for nucleic acid-based therapeutics, because every cell that you transfect will be a factory of your protein that you want to express. And therefore, the more producer cells we get, the more cells that we transfect, the higher the antibody output will be. And there are very well-established delivery systems for efficient delivery to liver hepatocytes, which are lipid nanoparticles. We usually express the antibody, if it's a full-chain antibody, on two distinct messenger RNA sequences. One messenger RNA is encoding the heavy chain of the antibody, and the other one is encoding the light chain. And then you usually mix these two messenger RNAs, encapsulate them into a lipid nanoparticle as a mixture, usually two of these against one of these in weight. And what you can then see if you transfect cells, that you get the functional antibody in the supernatant of the cells. In the next step, you then can encapsulate this in lipid nanoparticles. Here you have your messenger RNA in water, usually in low pH buffer, and you mix this with several lipids in a special composition that is usually dissolved in ethanol. You have an ionizable lipid, which is the key component that drives the efficacy and tolerability, and also binds to the messenger RNA during the formulation process and establishes the encapsulation of the messenger RNA. And then you have some other lipids that are principally stabilizing and giving a form and function to the lipid nanoparticle. If you inject this intravenously, for example, into mice, you can see an efficient delivery to liver hepatocytes that is driven by apolipoprotein E. Here you can see immunohistochemistry of the messenger RNA, and you can see that all of the liver hepatocytes are brown stained for the messenger RNA, whereas in the control you cannot see any brown stains. And there was no change in the liver histology. We were then interested in the antibody levels that you can reach with a messenger RNA. And here we had two different antibodies. One was encoding, one messenger RNA mixture was encoding for an anti-rabies specific antibody, and the other one for an anti-influenza B specific antibody. And you can see that we have a nice dose response in terms of antibody levels that we measure in the sera of the animals 24 hours after administration, depending on the messenger RNA dose. And more importantly, the antibodies produced within the animals were functional. For rabies, there's a VNT assay that is WHO qualified, and there is an WHO standard for protection, which is 0.5 international units. And you can see even with a low dose of 2.5 microgram per animal, we principally reach these levels. We were then also interested if we have a therapeutic effect. So here we challenged the mice with rabies virus in two different settings. One setting was the prophylactic setting, like in passive immunization, where we used the messenger RNA to produce the RNAntibody against rabies 24 hours prior the viral challenge, and a therapeutic challenge, where we first challenged the mice and then gave the messenger RNA encoding for the anti-rabies virus. And what you can see in both cases, the animals that got the anti-rabies mRNA therapy survived the viral challenge, whereas the ones that got the anti-influenza B specific antibody messenger RNA encoding LNP succumbed. So we were able, in principle, to express very high levels of antibody within messenger RNA therapy and also having functional protection against viral challenge. So with our research engine, we have a broad space available now for an antibody-based product development. We have a lot of more data also showing antibody treatment in oncological settings, with rituximab, for example, or bispecific antibodies. You can use this for antitoxins like snake venom or something like this. Passive immunization. It's a genetic concept, so compared to recombinant antibodies, one production facility can produce all of the different antibodies that you can think of just by switching out the open reading frame. And that also gives you a very easy translation from R&D to CMC. Whereas if you have a protein, you can sometimes hit issues doing upscaling, that you have agglomeration or solubility issues. I also showed that we can do various antibody formats like Fab fragments, single chains, B-specific antibodies, intrabodies, also camelid antibodies. And depending on the bioreactor that you use, if you, for example, inject into the eye, you would use the eye as a bioreactor, and then you have a high concentration of your produced antibody in the ocular space, for example. So with this, I hope I have give you a slight introduction into an antibody and RNA therapy, and I'm happy to take any questions if there's time left.
I have one quick question. Personally, how long does this mRNA actually stay in situ in the body?
Yeah, usually it's also depending on the sequence engineering that you do, how long you keep it in the translational machinery. Usually the half-life of a messenger RNA is between 6 to 48 hours.
Thank you. And there's one question. Sorry?
Yeah, the pharmacokinetic will also depend on the exposure of your protein, the half-life of your protein.
Okay. Yeah. Perfect. There's one more question from Zara Al-Ahmadi, and she's asking, what are the benefits of this technology compared to using purified recombinant antibodies? Is this more important to some diseases compared to others?
Yeah, it depends principally. If you, for example, inject into the eye, you would have a constant production on site in the tissue and constant exposure. And you usually every time enter the therapeutic window from below, because you start at 0 and you have then accumulation because you have a constant expression of your protein. From a recombinant protein therapy, you usually go to the upper end of your therapeutic window to keep the antibody level as long as possible in the therapeutic window. Here you are usually running into solubility issues, have microaggregation that also can be then immunogenic. So I think depending on your type of application, an RNA antibody therapy can be very good.
Oh, there's a lot more questions coming up, but I'm very sorry we have not much time, and I would like to actually continue with Dr. Don Parsons and the rapid deployment of messenger RNA vaccine for pandemic response, which is something that's really acute right now. Thank you very much. If I will try to get these questions to Dr. Baumhof, and hopefully he can answer them offline.
Yeah.
Thank you.
Really. Thanks. Bye.
Bye.
Thank you, Karin. Can you see my screen?
Yes, we can.
Great, thank you. Good afternoon, everyone. Thank you for the opportunity to present today at Cliniam. It's a great pleasure. My name is Don Parsons. I'm Vice President of Early Technical Development at Mrna Therapeutics. I'd like to tell you a little bit about Mrna's efforts to develop and deploy mRNA-1273, which is an mRNA-based vaccine against SARS-CoV-2. I'm going to reflect a little bit on where Mrna was as a company at the end of 2019 and how we've changed in the last 9 months as we've attempted to respond to the emerging public health crisis. I'll then speak a little bit about how the company has positioned itself, both technically and from a strategic perspective, to respond quickly to an emerging pandemic threat. I'll then spend a few minutes discussing a few of the many challenges which we were facing as we moved beyond our initial response, and focus on a couple of strategies in the CMC space that I felt were important to our progress. And then I'll close by speaking briefly about some of the many challenges that remain. See if I can advance the slide. Sorry. There we go. So entering 2020, Mrna was beginning our 10th year of operation. We had about 900 employees, about 400 of those were in CMC-related functions. We had 12 ongoing clinical trials, of which 2 were in phase 2, and we were working very hard to prepare for our first phase 3 trial for our vaccine candidate against cytomegalovirus. While we were still a young company, we'd enrolled a total of about 1,700 patients across all of our trials to date, and we'd established an internal GMP manufacturing facility that was supplying our clinical trials. Importantly, we'd scaled our manufacturing processes to approximately the gram scale of mRNA. 9 months later, a lot has changed. We've almost doubled the number of employees in CMC functions. We were able to supply the fastest ever phase 1 start of a vaccine, and as of last Friday, we have completed enrollment in our 30,000 patient phase 3 study. We've executed agreements with key manufacturing partners in the US and Europe, which will enable the production scale that we need for global supply of the vaccine, and we've been able to scale our manufacturing processes very significantly. But while a lot has been accomplished, there are many challenges that lie ahead. We were very focused in the first quarter of this year on enabling rapid clinical evaluation of a vaccine candidate, and we were able to produce clinical supply quite quickly once the antigen had been discovered. That rapid response is, of course, a critical capability in the context of a pandemic, and a capability we had been focused on for quite some time prior to COVID. The intrinsic speed of a vaccine design using mRNA is something I think that is well understood, but from a CMC perspective, it was really our investment in personalized medicine which was vital for rapid response. Our personalized cancer vaccine program laid the groundwork for mRNA-1273 by creating a CMC infrastructure that was specifically designed around speed from antigen design to manufacturing of clinical supply, by creating a highly efficient and coordinated manufacturing process and business processes to match that, and this was critical to achieving rapid phase 1 start. Another critical capability for us was an established in-house GMP manufacturing facility. While not common for emerging biotech companies, internal GMP manufacturing was vital for us because we had control of the facility's priorities, and we were able to respond with the urgency that the crisis demanded. A corporate focus on vertical supply chain integration to secure supply of complex critical materials was also impactful. A complex product like an mRNA vaccine has naturally a very complex supply chain with many critical materials, and spooling that up quickly could be a complex endeavor. Because we were either directly controlling supply or working in very tight partnership with suppliers, we were able to make this happen. And I think without this control, we would not have been able to execute so quickly. While we were able to use our existing capabilities to enable a rapid phase 1 start, there were many challenges that we needed to address as we moved toward later development and licensing application. One challenge was cultural. Over the past 5 years, we've worked very hard in the CMC space to enable the rapid clinical evaluation of multiple candidate products across a wide range of therapeutic areas, and we've become expert at rapid progress from proof of biology to clinical entry. However, we had not yet launched a phase 3 study, and the needed shift for our CMC efforts to a laser focus on a single product, which we needed to understand very deeply indeed. Of course, a key part of that understanding was the need for major scale-up, approaching 500x in only a couple of months. And that scale-up needed to extend across multiple processes from raw materials to mRNA to lipid nanoparticles to drug product. That scale-up would not have been valuable without the manufacturing capacity to go along with it. And we needed to very quickly identify partners with global manufacturing presence to assist us. And of course, we needed to tackle all of this while we were operating during the pandemic. I'd like to spend a few minutes talking about 3 technical strategies which were, in my opinion, important enablers of our ability to rapidly respond to the emerging pandemic. All of these were long-term decisions which required commitment years before the pandemic struck, but all were vital to the rapid response effort. The first of these was a commitment to scientific understanding of our products and processes. While it seems an obvious statement that a rigorous scientific evaluation should be the goal of any R&D effort, in the context of an emerging biotechnology company, the ability to devote resources and focus efforts on a long-term objective is not always straightforward. Without this commitment, though, the rapid scale-up of a complex product would have been very difficult to achieve. That vision was also important for my second point, which is the need to define processes with scaling explicitly in mind. Focus on good enough processes with the capability to supply early clinical trials in order to devote time and resources to other priorities is a natural temptation when those trials are the challenge in front of you. But having the discipline to design for the long term was ultimately vital when rapid scale-up was unexpectedly needed. Finally, I'd point to our approach to supply chain creation, which includes a focus on vertical integration. Any product as complex as an mRNA vaccine inevitably has a correspondingly complex supply chain. And it would be an inevitable thought to consider focusing on the core mission of an R&D organization and allowing specialist suppliers to assist in their areas of expertise. The complex supply network emerges from that approach, though, can be quite fragile, and when suddenly stressed, can collapse. I'd credit our internalization of much of that supply chain as a key enabler of our rapid response. I'd like to close by acknowledging that while we have overcome many hurdles over the past 9 months, many challenges remain, and I'd like to focus briefly on 3 of these. One will be the opportunity to discuss with regulators from many countries a technology which they are presently gaining familiarity. Our ability to demonstrate proper control and understanding of our products and processes will be underpinned by scientific understanding, which again underscores the value of long-term scientific focus. One particular area which will be a topic of discussion will naturally be product comparability. The ability to demonstrate that a complex product like mRNA 1273 is appropriately consistent across scale, across time, and across manufacturing sites will be vital. I point to a couple areas that I think will help us in this regard. And the first is process complexity. In comparison to many product modalities, and with appropriate diligence in process design, mRNA can be produced using processes which are relatively straightforward and conventional, and thus can be well understood and controlled. Naturally, we've sought and implemented robust process control strategies, but I'd additionally point to sustained and substantial investment in advanced analytical tools as critical for thorough product characterization. Finally, I'll note the global distribution of a temperature-sensitive product as an important challenge for us. I think it's been well appreciated that mRNA is a thermally labile material, and this has made product stability also a focus area for us. And we've been able to design a product which is stored long-term at -20 degrees. Obviously, such a storage condition will require careful and rigorous cold chain management, but this condition does fit within the existing infrastructure for many geographies, and I think that's an important enabler for us. I hope I've been able to paint a picture of some of the areas that have been important in undertaking rapid development and design of a vaccine candidate. But all of the technical considerations and capabilities would have been inadequate without the people at Mrna. So I'd like to conclude by acknowledging all of my colleagues, including those who have not worked directly on mRNA 1273, as well as folks who have kept focus on many other areas. They've put in many long hours over the past 9 months and have approached an array of challenges with unquenchable determination. It's our collective hope that we can contribute meaningfully to the fight against SARS-CoV-2. I'd like to acknowledge also our partnerships with NIH and BARDA, which have been vital to progressing development of the vaccine, and of course, the patients and healthcare professionals who are a very vital part of the mRNA 1273 journey. Thanks very much for listening, and I'd be happy to try and answer any questions.
Thank you, Don, for a very, very inspiring talk. And yes, it does take a village often to get actually things done. So we have one question. How long have you been able to sustain good anti-SARS-CoV-2 titers in treated individuals? Are you planning to perform real virus challenge with vaccinated individuals as planned with other vaccines in the UK? Do you detect robust CD8?
I don't think we have those type of challenge studies currently as part of our clinical plan. And I think we're still in the process, obviously, of evaluating the duration of immunogenicity.
Yes, I also would like to mention that there's a debate lounge 1 where we can also meet afterwards and look at the questions that we couldn't address in this time that we have. So with this, I would like to turn over to Dr. Steve Kenner for next generation gene editing technologies for allogeneic immune cell therapeutics. Steve?
Thank you. Thank you for inviting me to present today on Caribou's next generation gene editing technology. And we're applying this technology specifically to generate allogeneic immune cell therapeutics. So the idea is that, and before I start that, just to show you that I have one disclosure. So the idea is that because we're generating CAR T cells that are derived from a healthy donor, and then we're providing those to a variety of different cancer patients, the immune system of the host or the patient is going to clear it pretty quickly. And a couple of things are going to happen after lymphodepletion in the patient to make room for the graft. The cells will proliferate and then quickly become exhausted. And part of that is because they're going to express checkpoints that they could engage the ligands. They'll also differentiate into effectors. And then eventually the host will clear the graft. So we're taking 2 different approaches to address this, to increase the persistence of these cells. And one is to disrupt the PD-1, PD-L1 axis. There's been a lot of benefit from antibodies to these agents. And so our idea is to knock it out in the CAR T cells in order to take the brakes off the process of exhaustion. The other approach is immune cloaking, and that idea is to take off all of class 1 so that the CAR T cells cannot present antigen to cytotoxic T cells of the patient, but then reintroduce a minor antigen called HLA-E. And the idea there is that it's a ligand for a checkpoint on natural killer cells. And so we'll have the benefit of both the T cell side and the NK cell side of clearance in the patients by making these modifications. So to show you Caribou's pipeline, we have 4 products that we are advancing. CB10 is the most advanced, and that one is targeting CD19. We're knocking out, of course, the T cell receptor in these cells to prevent graft versus host disease in the patients. And we're also knocking out the gene that encodes PD-1, and this will be for relapsed refractory B-cell non-Hodgkin lymphomas. This is the product candidate that I'm going to be talking about today. There are several others, one targeting BCMA for multiple myeloma and a couple of others that I won't talk about today. Our platform is a next generation CRISPR technology. So everyone is familiar with Cas9 and all RNA guides. But what we've done is we haven't modified the enzyme, but we've modified the guides. And the reason that we do that is to improve specificity, so that when we generate a double-stranded break with this technology, we're not ending up with many double-stranded breaks in off-target sites. So the idea is that we're introducing DNA into the backbone. So it'll be, rather than an all RNA guide as naturally is generated, we'll have a mixture of RNA and DNA in the backbone. And we call these hybrids CRISPR hybrid RNA DNA, or chRDNA for short. And the application of these chRDNAs is flexible. We can use it with different type 2 enzymes, including Cas9 and Cas12a. And the company has exclusive rights based on a number of patents. So if we employ these chRDNAs and ask the question, if we compare it to an all-RNA guide, is the on-target editing similar? And so these data here show you that if we take the enzyme, mix it with a guide of choice, and then nuclear affect that into primary human T cells from blood, that if we look at on the left in Cas9 platform, regardless of the sequence that we use, we can compare that head-to-head, an all-RNA guide in the lighter shade to a Chardonnay in the darker shade. And these guides are the exact same sequence. The only difference now is that the Chardonnay has some DNA in the backbone. We get the same level of on-target editing. So this is based on deep sequencing at the site. And so we're able to retain very closely the on-target editing. So now the question is, okay, if the editing is similar, is the off-target profile different? And that's really what we're trying to achieve with this technology. And the way that we evaluate that is a two-part system that was developed by the company a few years ago. And the idea is that we take unedited cells, isolate genomic DNA, and put the genomic DNA into a test tube together with the enzyme and the guide of choice, and allow it to digest at will. So it's completely unbiased biochemical assay. Then we capture all of the potential double-stranded break sites by a combination of PCR, barcoding, and deep sequencing. And then on part 4 in the upper right, you can see we've developed sort of a map of all the potential double-stranded breaks that occur. So this is just showing you the number of breaks that occurred, but we also know exactly in the genome where they occurred and how many mismatches between the guide and the chromosome there were. So if you look at the upper right, this is just by way of an example. This isn't meant to be relevant to one of our product candidates, but just to show you that a CRRNA, which is an all-RNA guide, or CRISPR RNA, induces many double-stranded breaks. The more we add to the test tube, the more breaks we see. But the Chardonnay produces fewer. So now we take this sort of menu of places to look, perform the experiment as I showed a minute ago, transfect or nuclear effect in our guide and enzyme into the cells, and then evaluate based on all the sites we learned from the site-seq assay. So a summary of that data is shown here. And so just to show the power of the technology, we used a sequence that's known to be highly promiscuous, meaning that in addition to the on-target, it also induces many off-targets. So if you look at the far left using the Chardonnay Cas9 platform, we took that known sequence to AAVS1, put it into the system and saw about 90 to 95% on-target editing in the dark blue color. Next to it, you see 3 off-targets. So this is, not desirable because if you're generating very high levels, 80% of an off-target, that could lead to translocations later, which could be problematic for the product candidate. But if you look now to the right, head-to-head, we also looked at a Chardonnay of the same sequence, but now with RNA and DNA in the backbone. The on-target editing is equivalent to the all-RNA guide, but there are no off-targets. So the power of the platform can be applied across all sequences. We generally never see any off-targets with the Chardonnay platform, but we generally do see some off-targets with the all-RNA guides, as you can see. And the same was true with the Cas12a platform. If we applied it there, again, if we take a promiscuous guide, we can generate many off-targets and we can eliminate them with the Chardonnay platform. So this is great because now we can generate a single double-stranded break to induce a mutation in a cell, like knock out a gene, disrupt a gene. But what if we want to make multiple edits? And as I described in the beginning in our pipeline, we're making multiple edits in our product candidates. So the team set out to address that as well to reduce the likelihood of translocation. So by way of example, on the left, if we were to knock out a gene in chromosome 14, say the TRAC gene, and then another gene, PDCD1 in chromosome 2, that could lead to these translocated chromosomes as shown in the lower left. These would be stable centromere-containing chromosomes, and we could PCR across those cut sites to prove that there were translocations, and we did just that. If you look in the box in the center, you see the editing efficiency of each of the 2 genes independently that came along with 3.5% translocation. And this is consistent with what's been seen in the literature using either CRISPR technology or many other gene editing technologies, such as TALENs. So the team set out to reduce the likelihood of this, and we modified the nucleafection process, the delivery technology. It is a trade secret of the company, but it doesn't change the reagents. It's really just the nucleafection system, the platform. And so you can see that the gene editing efficiency of each gene went up somewhat, but the translocation rate went down in this particular case about 35-fold. So together with the chRDNA and this delivery approach, we should have a very clean profile with respect to the genomic integrity of our product candidates. So the product candidate I'd like to talk about today is called CB10. It targets CD19. The idea is that we're going to introduce this CAR into the T cell receptor alpha constant locus to knock out the T cell receptor to reduce the likelihood of GVHD in the patient. And we get enough of that CAR DNA into the cell by infecting that T cell with AAV6 that contains a plasmid expressing the CAR. In addition, we're knocking out PD-1 to reduce the likelihood of exhaustion. And this particular product candidate uses exclusively Cas9 chRDNA reagents. So we generated the cells and showed that they have in vitro functionality, as we would expect. On the left, it shows that they have dose-dependent cytotoxicity against tumor cells. There's 3 different varieties shown there in terms of types of leukemias or lymphomas that we're looking at. And also they induce the expression of a variety of different cytokines. 2 are shown here. In addition, we included something that's hard to see, but something called CAR-KI. So these were CAR T cells where we introduced the CAR into the track locus, but we didn't knock out PD-1. Just to understand what's the role of the knockout of PD-1 in exhaustion, but these assays were run in 24 hours, and so we couldn't see the long-term persistence of that knockout. And so we set up a xenograft where we could look very long-term at the effect. And so here we could do the head-to-head comparison in this xenograft, evaluating the effect of the knockout. And so this is a CD19 positive, PD-L1 positive tumor, and we established it orthotopically to represent what would happen in a patient. And we let it grow for 23 days to ensure that it was highly metastatic within the mice, also to reflect the condition that a person would experience with their cancer. And so these tumors could be lit up by luciferase and imaged. And so what you see on the far left is what we call day -1 or 23 days of growth of the tumor. And you can see that the mice have very high tumor burden. We then treated with either PBS, a non-targeting CAR T cell where the CAR is introduced into the track locus to knock out the T cell receptor, but that CAR has no antigen binding capability, so no scFv or antibody fragment. We then utilized allo CD19 CAR T cells, or our CAR-KI. This is where we put the CD19 CAR into the track locus, but did not knock out PD-1. And then CB10 that has the PD-1 knockout in addition to the CAR. And so you can see 2 weeks later that there is a high level of metastasis within the 2 negative control groups, but both the allo CD19 CAR T or CAR-KI group and the CB10 group showed full eradication of that tumor. And then we took it out to day 108, and you can begin to see that all of the animals in the negative control groups had expired. Half the group that received the regular CD19 CAR T, they expired, and the other half were highly metastatic, and all of the mice in the CB10 group were still alive with very low tumor burden. And then we were able to let this study go out to 160 days, and that's shown in the survival curve here on the right. By day 135, all of the animals in the allo CD19 CAR T or CAR-KI group had expired, but we'd only lost 1 mouse in the CB10 group all the way out to day 160. So we feel that this is not only significant, with respect to statistics, but also clinically meaningful, because a patient that we'll be targeting has a lymphoma where maybe 50% of the patient's tumors express the ligand PD-L1 that would engage with PD-1 on the T cells, but also the microenvironment contains a lot of ligand. So immune cells express PD-L1, and also the epithelium of the lymph node also expresses the ligand. So That's why in the CD19 CAR-KI group, where we did not knock out PD-1, they expired quicker. So we think that this will be meaningful in the clinic. And speaking of the clinic, we're also going to be looking at B-cell non-Hodgkin lymphoma patients. And so we wanted to address CB10 in models of those two diseases. Two of those diseases. And DLBCL and MCL were two of them that we evaluated and showed improvement in survival. We also looked at safety of CB10 by asking the question, in mice that do not harbor tumors, if we infuse them, does it cause any kind of clinical parameters, loss of body weight, or infiltration into tissues? And so we also compared it to T cells that were unmodified from the same donor that we prepared the CB10 from. And you can see that only the wild type T cells showed any kind of clinical parameters, loss of body weight, or infiltration into tissues, while CB10 did not induce any of these effects. So this, confirmed to us that we're not going to see GVHD with this product candidate. This is the process of how we make these cells. This is done in a cGMP lab external to the company. And we include a step right before we cryopreserve them, shown on the right, where we use anti-TCR microbeads to get rid of any possible TCR positive cells remaining. Gene editing, although high efficiency, can be upwards of 95 to 98% efficient. We're not knocking out 100% of the T cell receptor in the population of cells, so we need to get rid of them. And this process eliminates them down about 2 orders of magnitude lower than the starting material, which is about an order of magnitude lower than what the agency requested of us. So the trial that will be starting before the end of this year is called ANTLER. The indication is going to be relapsed or refractory B-cell non-Hodgkin lymphomas. We're opening it up to 7 different subtypes that are shown there. And the main reason is that we want to do a basket trial because we don't know where we're going to see the best efficacy. And many of these subtypes remain underserved by CAR T therapy today. This will be targeting adults who have failed at least 2 lines of chemoimmunotherapy. And we intend to exclude any patients that had any sort of prior CD19 targeted therapy in case that debulked their tumors and led to CD19 negativity. We're going to use a standard 3+3 design. The intent is to find a recommended phase 2 dose. Once we do, we will then go back to either 1 or 2 of the subtypes shown at the top and perform an expansion phase on those types of patients to identify potential for efficacy and further safety. And as I mentioned, we'll be enrolling before the end of the year on this trial. So just to summarize, I just want to reiterate that we have this flexible platform that is highly specific and is an improvement over the first generation CRISPR technology. And we have multiple ways to deal with genomic integrity that we think will make a safer product for patients. Our pipeline includes many different types of allogeneic cell therapies. And today, just really to reiterate that we're taking this one approach to try to improve the long-term persistence, particularly of the anti-tumor activity. And then later we'll be looking at persistence in circulation. So mostly I want to thank the folks who did the work, and now I can take any questions.
Thank you very much, Steve. So I have one question, and that would be, do you anticipate any kind of side effects from that therapy like we see in the normal, autologous setting with cytokine storms and so on and so forth?
Yes, we do. I think that all of the therapies seem to induce that to some rate. What we're hoping is the fact that we've knocked out PD-1 and we see this accelerated, amplified anti-tumor activity that we saw in the preclinical model, that potentially we could use a lower dose for debulking the tumor, and that might reduce the, sort of the prevalence of CRS, but I would anticipate we will see some. I don't think GVHD will occur, and our competitors haven't seen it either, so I don't think that's going to be problematic. But we certainly will be paying attention to CRS, ICANS, which is the neurotox, as well as neutropenia, which is problematic for infections.
And then there's one question from Magdalena, and she's saying, can you please comment on challenges related to nucleofection with CRISPR-RNP and AAV transfection in your CB10 product? What is the cell recovery at the end of the process?
Right, so cell recovery is actually great. It's just that we don't want to grow the cells too long. Once they recover after all of that, you just feed them IL-2 and they continue to proliferate wildly. But if you let them grow too long, then they begin to shift towards effectors. And we find that when we evaluate them in our preclinical models, that the effectors don't work as well as if we keep them in the central memory, which means don't letting them grow too long. So we basically stop the process short. And, of course, we don't get as many doses per batch that we would love to have, but we want a better product. And so they do recover, but we have to stop them early to prevent that from happening.
I didn't catch this on your graph. How long was your process?
Right, I'm not allowed to say.
You're not allowed to say.
But I appreciate the questions.
That's fine. Cool. That was very interesting. And with that, I have to go on now to Dr. Brett Monia. Exactly. And he's going to tell us on recent advances in the antisense technology.
Hello, everybody. Can you hear me? Hope so. So it's a pleasure to be here today and provide an update on antisense technology at Ionis Pharmaceuticals. I can't advance for some reason. It's not advancing. It's not advancing. No.
Maybe you can just.
I could make it work. I'll just use that. Okay, here we go. I'll just use that. So here we go. So some key take-home messages. Antisense technology is here and now really delivering on the promise that was originally sought many decades ago when it was first conceived, that by targeting RNA with oligonucleotide-based drugs, we can really build a new platform for drug discovery, with several products now on the market and a rich pipeline of new drugs to reach market anticipated for next several years. In addition, the technology continues to advance forward with investments in research and medicinal chemistry and new routes of delivery. And you'll see some of that during the course of my presentation. So antisense technology, I think everyone is aware of how it works. We're targeting RNA through Watson-Crick hybridization mechanisms of action, in which we bind to the RNA using chemically modified oligonucleotide-based drugs through Watson-Crick hybridization. These are drugs between 15 and 20 nucleotides in length, single-stranded. And upon binding to the RNA, they can do several things, use various mechanisms of action to either decrease the production of a toxic protein or a toxic RNA, or increase the production of a protein that's lacking in loss of function diseases. We have demonstrated that we can use and apply antisense technology and affect gene expression in a broad range of organ systems and cell types. And we can do so through the utilization of medicinal chemistry, which can traffic oligonucleotides to different organs and cell types in certain ways, following subcutaneous or intravenous administration. Or we can deliver these drugs to organs of interest through local routes of delivery, such as inhalation delivery for pulmonary diseases, or intravitreal administration for ocular diseases, and in addition to the CNS through direct administration to the CNS or intrathecal administration. And even oral delivery is now, we believe we're on the verge of tackling commercially viable oral delivery using certain types of chemistries that we've developed as a once-a-day tablet. Today, there are three, from Ionis, there are three antisense medicines on the market. They target genetically caused rare diseases, Spinraza, for all forms of spinal muscular atrophy. Tegsedi is now on the market for hereditary TTR amyloidosis in patients with polyneuropathy, and Waylivra is now approved for a genetic disease called familial chylomicronemia syndrome, or FCS. In addition, today we have 5 phase 3 studies in progress using this platform, and a 6th phase 3 study is expected to begin later this year. These are for rare diseases, such as for Huntington's disease and ALS, and follow-ons to Tegsedi and Waylivra using new chemistries, and in addition to very large indications, such as for Lp(a)-driven cardiovascular disease, pelacarsen, which is in a cardiovascular outcome trial involving more than 8,000 patients today. Our pipeline is nearly 50 drugs in development across from phase 1 through phase 3 development, and we're expecting quite a number of new drug applications through 2025. The leading franchises, the large franchises for in our pipeline are cardiovascular and neurological diseases. This is what our cardiovascular and neurological disease pipeline looks like today. We have 9 or more drugs in each of these franchises, ranging from phase 1 through phase 3 development. As you can see, we have several partners that we're working with in the development of these drugs for cardiovascular and neurological diseases. And there are many drugs that we're developing ourselves and plan to bring through phase 3 to the finish line and to commercialize. So what I wanted to focus on today in the time I have allocated is on neurological disease pipeline, because I believe that the breakthroughs we've made in neurological disease drug discovery and development make this platform a premier platform and a leading platform in the treatment of neurological diseases, which of course was led by the development and the successful commercialization of Spinraza for all forms of spinal muscular atrophy. You know, we all recognize the fact that the Human Genome Project has provided tremendous benefit to our understanding of biological causes of biological diseases. And in particular, neurological diseases is an area where the progress has been really enormous. The genetic causes of so many neurological diseases have been laid out and understood at the molecular level over the last number of years. And therefore, what is needed are new platforms for drug discovery to take full advantage of the causes of these neurological diseases to help patients where there's really no treatment options available. Large pharma using traditional drug discovery platforms have struggled. I think it's fair to say, using small molecules or monoclonal antibodies, for example, to treat these genetic causes of neurological diseases. But with the advancements we've made at Ionis developing and optimizing the antisense platform for the treatment of neurological diseases, we think that we are on the verge of really delivering many new drugs for the treatment of all kinds of neurodegenerative diseases. The key step that we made, which took 8 to 10 years of optimizing through medicinal chemistry and understanding distribution and pharmacokinetics and safety and tolerability, is the route of delivery shown here, an intrathecal delivery of oligonucleotides to the central nervous system. Oligonucleotide drugs do not cross the blood-brain barrier, so we need to deliver them directly to the central nervous system. We have a lot of experience now with this route of delivery, preclinically as well as in the clinic. Spinraza has now been treated in more than 11,000 patients around the globe with remarkable efficacy, safety, and tolerability. And we have clinical trials now well underway for Huntington's, Alzheimer's, and Parkinson's disease. So what do we know about distribution in the CNS following IT delivery? Well, we know that it's very similar across species, from mouse through dogs, pigs, non-human primates, and in humans. We know that bolus injection, a single one-time injection, is we achieve greater delivery efficiency, broader distribution versus an infusion, a constant infusion route. And we know that these drugs distribute very broadly in the spinal cord and in the central nervous system, and affect gene expression very potently in essentially all relevant cell types in the CNS. We know that the highest concentration is in the gray matter, in brain and spinal cord. And as I mentioned, we can show efficacy, activity in all the cell types that are listed on this slide. We also know that, and have demonstrated that these drugs last for a very long period of time in the CNS, for 3 to 6 months duration of effect, so that we only have to give a bolus injection once every several months to achieve consistent and reproducible efficacy. We believe we are on the verge of cracking the code for neurodegenerative diseases. We have drugs now on the market, in the clinic, or soon to be in the clinic for all these therapeutic areas for neurological diseases today. You can see that essentially every aspect of neurological diseases that you can think of where there's a large unmet medical need, we're in it. Whether it's rare diseases such as Huntington's disease or amyotrophic lateral sclerosis, or rare disease, or broad diseases such as Alzheimer's disease and Parkinson's disease and so on, and even diseases like prion disease, where there's absolutely significant unmet need for this lethal type of disease. We're also moving into new areas such as severe chronic pain and neurodevelopmental disorders, and even psychological disorders are now on our menu of things that we're going to be moving into. So in the time I have, I wanted to give one story on how we're applying antisense technology for the treatment of neurological diseases. And this is and this story is about a drug called tofersen, which is now in phase 3 development for the treatment of a genetic cause of amyotrophic lateral sclerosis, or ALS. ALS, of course, is a lethal disease. It's a severe disease characterized by motor neuron degeneration, resulting in paralysis, respiratory failure, and ultimately death in all cases. It's rapidly progressive, typically 3 to 5 years from symptom onset to death. And we know that there are many different causes of ALS, but they can be classified into either genetic causes or non-genetic causes, or sporadic ALS or broad ALS. Broad ALS, of course, affects the majority of patients suffering with ALS, but there are many different causes of genetic causes of ALS as well. We have programs in both genetic and broad ALS for the treatment of these diseases in the clinic today. But I want to focus on this story because it's the most advanced ALS program in our pipeline today. This is a drug, as I mentioned, called tofersen, which targets a genetic cause of ALS due to mutations in a gene called SOD1, or superoxide dismutase 1. We know that this disease is as a result of mutations in SOD1 due to the misfolding of the protein. It's not due to any contributions of the enzymatic activity of SOD1. It is entirely due to a misfolding of the protein causing aggregates, resulting in neurodegeneration. We have shown in animal models that we can reduce SOD1 in the CNS by intrathecal delivery and improve survival in ALS models. We've also shown that in the clinic that we have early signs of efficacy, and we are now in phase 3 development with this drug, which I want to show you a little bit more on right now. When we advanced tofersen into phase 1/2 study, we did obviously a dose escalation for safety initially, using a single ascending dose regimen in which patients with SOD1 ALS with symptoms were treated initially with a single dose at increasing dose levels. And then that followed, after safety review, into a multiple ascending dose regimen in which patients were treated every month at weeks 1 to initially at weeks 1 and 2, and then every month thereafter at week 4, 8, and 12. A 3-month study, if you will, and then patients were followed for 12 weeks following completion of the multiple ascending dose study. What we showed in this study was that we were achieving concentrations of tofersen in the CSF that were dose dependent as expected and in line with what we expected to achieve to produce reductions in SOD1 that we were predicted to demonstrate efficacy in patients with SOD1 ALS. And indeed, we saw very nice dose-dependent reductions in SOD1 ALS, in SOD1 protein levels in the cerebral spinal fluid in patients. That was also dose-dependent. The tolerability profile and the safety profile was very attractive. We selected 100 milligrams, the highest dose, to go into the phase 3 study. But before I get to the phase 3 study, I was jumping ahead a little bit. I wanted to show you this data, which was really giving us confidence in what we were expecting in the outcome of the phase 3 study. This slide shows early signs of encouraging clinical efficacy in this 3-month study in which we evaluated tofersen in patients with SOD1 ALS. On the left side is a rating scale called the ALS Functional Rating Scale, which is a measure of all aspects of neurodegenerative disease progression in patients with ALS. This is a validated instrument that is accepted by regulatory authorities for approval. And on the right is lung function. Remember, these patients succumb eventually to respiratory failure. In blue in these graphs is placebo-treated patients over the 3 months of treatment with placebo during this study. And in orange are patients treated with tofersen. In the solid lines is the entire population of patients. And what you can see is that patients treated in the solid lines shown in orange with tofersen compared to placebo in the solid blue lines, that there's a strong trend favoring stabilization of disease in patients treated with tofersen. What I also want to draw your attention to are the dotted lines. Depending on the mutation in the SOD1 gene that patient may have, you can have a form of SOD1 ALS that is referred to as rapidly progressing. Mutations that cause rapidly progressing disease obviously result in disease progression that's much faster than other forms of SOD1 ALS and result in death much sooner than the typical SOD1 mutations. Shown in the dotted lines are the patients with rapidly progressing SOD1 mutations. And there you can see a real differentiating effect after only 3 months of treatment in patients treated with tofersen on the functional rating scale on the left and in the lung function on the right. And of course, we expect tofersen to be efficacious in all forms of SOD1 ALS. The reason why we can see this differentiation in rapid progressors in placebo versus treatment is because it's only a 3-month study, and those patients in placebo progress much more rapidly. So tofersen is an investigational medicine that we have generated encouraging results in a 3-month phase 2 study in patients with SOD1 ALS. As I said earlier, we selected 100 milligrams for the phase 3 dose based on the efficacy demonstrated with 100 milligrams in the phase 2 study after only 3 months of treatment. And that phase 3 study called VALOR is now well underway. And this is what the design of that phase 3 study looks like. 100 milligrams dosed monthly to patients, approximately 99 patients with SOD1 ALS. We have a placebo group and a treatment group, of course. And the treatment period is 6 months, so twice as long as the phase 2 study, so we can really understand the progression and see the differentiation in all forms of SOD1 ALS compared to treatment. And then patients will be rolling over into an open label extension following completion of the 6-month study. I forgot to say that that study is now nearly fully enrolled, and the phase 3 results are expected to read out next year. And we expect this drug to be the first of several drugs from our pipeline for the treatment of all forms of ALS. SOD1 ALS data expected, as I said, next year. We also have a phase 2 study in progress targeting another genetic form of ALS, targeting a gene called C9orf, where the mutations in C9orf cause ALS due to those mutations. We just initiated a phase 2 study in sporadic ALS, non-genetic ALS, by targeting a gene called ataxin-2, and that study is now underway. And we're planning to start a phase 3 study next year for a third genetic form of ALS, a fourth total from our ALS pipeline, targeting mutations in the gene called FUS. And then finally, I'll close.
It's Karin. I'm sorry. We kind of have to wrap it up.
Oh, okay. I'm
getting late.
up right now. Yeah.
Thank you.
So that's my concluding slide. We think that the technology is delivering now significant values to patients, and we're very excited about neurological diseases, but also our entire pipeline. Thank you.
Excellent. Thank you. There's 2 questions, one from me and one from Michael Keller, which is actually the same question, saying, have you looked at alternatives to the intrathecal administration, meaning in the spine, that is cumbersome for patients with chronic disease? So what I'm thinking of is also the Alzheimer's disease patients that have to come back and get an injection into the spine, and they might not really want it. Is there really a improvement in Alzheimer's for the people that get this kind of injection?
Yeah, that was actually the slide I skipped. We're making new advancements in the technology for neurological diseases, and it's coming in 2 forms. One is that through medicinal chemistry, we think we're going to be able to get to annual dosing, once a year dosing, intrathecal. And then second, we're working on new chemistries that will actually cross the blood-brain barrier. And that's further out. We think that we'll have chemistries for once a year dosing coming very soon. And we're actually expecting to develop a drug to follow on to Spinraza with that chemistry. And then we're working on the blood-brain barrier.
Excellent. Thank you very much. And that gives us a lot of hope to those people with these horrible neurological diseases. Thank you very, very much. I'm now turning it over to Magdalena Oberjanik-Voigt, and she will tell us a little bit about the innovative cell and gene therapies for non-oncology indications. Magdalena?
So hello everyone, and thank you to the organizer for giving me an opportunity to actually talk to you today. So I'm actually a drug product leader in Novartis, located in the technical research and development within global development, drug development team at Novartis. And today I will be talking about other cell and gene therapies, not the CAR T therapies, as we have presented a lot already as Novartis on this. I want to put your attention today on other indications, such as non-oncology indications. Please allow me to start with this disclaimer. This presentation is based on publicly available information, and slides are intended for educational purposes. If you want to share them, use them, please contact me upfront. Basically, the view and opinions expressed in this presentation are those of the presenter and do not necessarily reflect the official policy or position of Novartis or any of its officers. My talk today will be composed of 3 main parts. I will bring a little bit of introduction to cell and gene therapies, and then I will talk briefly about development approaches and challenges in 2 therapies: cell-based gene therapy, showing you a therapy that we developed for sickle cell disease, and the second one, adeno-associated vector therapies for retinitis pigmentosa. Before I go into the topic, please let me tell you a little bit more about what cell and gene therapy is. In high level, cell and gene therapy can be classified in 2 different types: in vivo or ex vivo. In vivo therapies are those that basically bring the genetic information directly to the patient. We have in our portfolio at Novartis 2 products like that: Luxturna, that is intended for supraretinal injection, and Zolgensma for neuromuscular diseases. Ex vivo therapies are those that require a collection of the patient material, expansion of isolated cells, editing of those cells with genetic information, and bringing back to the patient. We heard today about other example of ex vivo therapy for allogeneic use. In the Novartis portfolio, we have Kymriah, which is actually autologous cell therapy, CAR-T based. Brief history of cell and gene therapies. The story started actually already in 90s. So it's nearly 30 years that we are hearing about cell and gene therapies. First trials were using retroviral vectors for editing either T cells or hematopoietic stem cells with different success rates. Due to the fact that retroviral vectors have tendency for inducing some oncogenes, society started to move towards more safe vectors such as lentiviral vectors. In 2015, 2016, we started to hear about first therapies coming to the market. Imlogic for cancer using modified oncolytic HPV encoding GM-CSF, and also Streamvel is one of the therapy using retroviral vector that started in early 90s development. We also have seen the first proof of concept for AAV in vivo therapies for retinal dystrophy. The great breakthrough came with the first approval of CAR-T therapies, and we as Novartis, we were the first to come to the market with our commercial product Kymriah, with the follow-up of Yescarta by Kite, and in second half of 2017, also Luxturna came to the market, which is also to treat subretinal diseases. To put cell and gene therapies a little bit into perspective, there are different types of therapies. We speak about viral-based therapies and cell therapies, and in the right-hand table, side of the table, you see actually viral vectors and cell therapies. Those are, regarding viral vectors, they are very small molecules, very small particles of the size of between 20 to, let's say, 300 nanometers, while cell therapies are much larger particles of a size of greater than 5 microns. What actually came with the first therapies that coming to the market was a difficulty with having no really clear regulations in place. If you remember from my previous slide, we actually had some first therapies already showing up in 2017. And at that time, both in the European market as well as US, we had only guidelines. So Novartis also had an excellent opportunity to contribute to development of these guidelines as well. Now I would like to start with the first case, first example of therapy. And here I will be talking about cell-based gene therapy for sickle cell diseases. The sickle cell disease is a pretty common occurrence with 300,000 cases per birth per year. Unfortunately, it is also related with quite high death rate of approximately 175,000. For time being, there is no clear curance for this disease. We have treatments such as hydroxyurea, transfusion, or bone marrow transplantation. Especially with the last one, connected with the difficulty to find sibling match. The life expectancy of this disease is approximately 40 to 50 years. And the cause of this disease is a mutation in beta-globin, which leads to the formation of disrupt erythrocytes. Erythrocytes starts to clog into the blood vessels and finally leading to chronic anemia, pain crisis, and last to multi-organ damage. So to find a cure for this disease is really significant. treatment. And at Novartis, we have developed a therapy that uses patient-owned cells, hematopoietic stem cells, CD34 positive, which are mobilized in patient, collected in the process called apheresis, then sent to the manufacturing site where hematopoietic stem cells are gene edited using CRISPR-Cas9 technology, expanded in vitro, formulated, forming a gene therapy that can be shipped back to the clinical site and infused to the patient. The whole process of the gene therapy development is focused on main four aspects. First is source material. Second, gene editing technology. Third, expansion process, and the last, formulation and fill and finish. Each of this process step is pretty critical to the final outcome of the formulation of the product. Let me focus today only on two main aspects, source material and final formulation. I will start to speak about source material because this is really critical part of the developing of good process. As you may imagine, the development process cannot use patient own cells. So many times we need to rely on healthy donor material. And in this situation, we speak about a different donor, access to this material. Another aspect is material quality and especially donor variability. And here in the slide, I want to show you also an example of leukapheresis variability, where we phenotype the different donor materials to see on the population of different cell types, such as B cells, monocyte, NK cells, or T cells. You see huge variabilities between 10 to 90% between different donors. So the first and the most important aspect that developers had to deal with is the variability of the material that enters process development. Another very important aspect is the source of the material and mostly the age of the material. We have been developing different products also, and having been testing different source material. And we realized that it's very important to develop the process in a way that can accommodate to different source material quality. As an example, I want to show you the quality of the product from umbilical cord blood that can vary from cell recovery between 20 to 80%, and that can bring a lot of debris into the process. How we approach the process development. So development and feasibility studies are mostly done using healthy donor material. And this is due to the fact that we have pretty relatively easy access and we can get this material pretty quick. It can also allow multiple repeats. However, due to the material donor variability, we have to develop the process using multiple donors. So we need to run multiple repeats of the process in order to find the most critical attributes, quality attributes that influence the process, and to develop process in a way that is really robust. To really confirm our process development and the process robustness, we have to perform confirmatory runs using small-scale model or patient material. And this may be very limited. So especially access to patient material might be hard. Therefore, many times we need to develop a small-scale model that we can test the process. And of course, especially if we want to select a new technology or compare the technologies between in a head-to-head study, we often have to approach in a split run. So taking the material and splitting in two and running in parallel. Only this way we can find the best process improvements. Another aspect for the source material that we need to deal with is formulation. Because of the access to the different donor materials, we use actually multiple suppliers to provide us the material, and often we do not have an impact on the formulation or anticoagulants that are used in the process. Another challenge is that many times we have to deal with only few 200 milliliters of the product, and if we want to develop the process that can accommodate to large quantities of apheresis, sometimes it's very difficult to find the best approach. We need to focus on the process already at the beginning to find the best parameters that can be scaled up easily. Another aspect is packaging type, or actually whether we can get the material in vial or bag. Cell therapies are mostly focusing on having a closed process due to the fact that we are not able to sterilize the material, and therefore it is better to get the material already in bag, because the bag can be attached to the process immediately and in an aseptic way. However, often we need to deal with the material that comes to us in a vial, and that brings another challenge to the process development. The last that has a critical impact on the quality of the material is the type of the material used for storage, and mostly the packaging material composition. Let me talk a little bit about formulation and fill and finish development. So I mentioned already that we have a high donor-to-donor variability, but actually donor-to-donor variability is not only related with the amount, or with the type of cells that we have, an amount of the cells that we have in apheresis, but also unique donor cell predisposition to expand in culture. To overcome that, we try to use multiple batches to confirm our formulation or fill and finish process. Another challenge to the fill and finish and formulation development is the fact that both cells actually cell process is actually under development, and analytics is also under development. Due to the fact that we have a high limitation in batch size, and especially important if we want to develop new formulation, we would like to, for instance, run design of experiment, we may not have enough material to evaluate all possible scenarios, especially in the scale up approach. So therefore, it is very critical to develop representative downscale model for formulation development activities. Regarding analytical methods which are under development, we may approach the process development in a way that we define the discriminative assays for formulation screens. We do not have to check, for instance, the phenotyping of our product, but it's critical to look on the cell viability and cell recovery, pH of our formulation, and impact on the process. And very important, what I want to mention here is the early definition of target product profile. We need to know very early on in development activities whether we speak about dose that is equal to one single batch, the one batch that is equal to one single dose, or we are speaking about dose as a container for autologous projects. It is also critical to us to know whether we want to have a fresh or frozen product. Let me go with a couple of next minutes into gene therapy for retinitis pigmentosa, which is a totally different gene therapy that we are developing at Novartis. Retinitis pigmentosa is an orphan disease, a genetic disorder, which with around 500 patients worldwide. It is caused by mutation in RLBP1 gene, and and there is definitely no cure for this disease. The progress of the disease already started in the childhood, and with the progressive loss of vision, especially peripheral vision, leading to so-called tunnel vision, or may even lead to complete blindness in the age of 40s. Novartis has developed a subretinal injection gene therapy using AAV vectors to treat this disease. However, I want to concentrate here on the process development, and in this figure, which I want to depict actually the main process steps of the gene therapy development. As you can already see at the first glance, this process looks totally different than the cell process I showed you a couple of slides ago. This process may resemble to you a standard biologics production process. So we can easily distinguish upstream processing part, downstream processing part, and fill and finish. Upstream processing starts with working or master cell bank vial going through cell expansion, plasmid transfection, then vector harvest, purification, and up concentration to formulation, fill and finish, and storage. So of course, the process development for this type of therapy has totally different approaches. First, we need to look on the colon selection and characterization. We, for instance, look at the population doubling time, passaging, and cell recovery. And this may be very difficult if we do not have representative small-scale model. But it is much more easier to introduce a representative small-scale model for this type of gene therapy compared to cell therapies. Another aspect for development of gene therapies, such as AAV-based, is a selection of the best culture type, whether we want to have adherent process or suspension process. Of course, a suspension process is ideal due to the fact that can lead to a higher or greater yield, but it's also much easier to scale up because the adherent process can be only scaled out, which is very laborious. For the culture type, we also speak about culture process optimization, seeding density, and optimization of parameters of culture. The next step is efficacy of transfection. So we absolutely want to have the best yield possible by optimizing the co-transfection process, and this can be easily done by plasmid ratio optimization or the concentration of DNA that is used in the process. The next step is DSP vector recovery and stability, and as you can guess, this is really the main struggle for the downstream processing. We are focusing on finding the best parameters to improve the impurities clearance, to have a full capsid enrichment. The main challenge on this aspect is, of course, to find the process parameters that can be scale up. The last part of the process development is fill and finish and vector stability, and here we want to have the best parameters to support product stability, because vectors are pretty sensitive to shear stress and pumping, but have also high propensity to absorption to the wall of containers, as well as to particle aggregation. And with that, I would like to summarize and conclude. Today I have shown you two different types of gene therapy. So one that was cell-based gene therapy, and the other that was viral vector-based gene therapy. Both seem to be connected with totally different challenges. The first one focusing on the criticality of source material, and especially access to this material, as well as quality, because those has significant impact on process development. And the development strategy needs to accommodate to attributes of starting material. Cell therapy is pretty expensive in development and labor intensive, because one batch leads most of the time for one dose, especially if we talk about autologous therapies. And another challenge in cell therapies is many manual steps that requires highly experienced and qualified operators. On the other hand, viral vector-based gene therapies seem to show a much more conservative development approach with clear focus on USP, DSP, and fill and finish process steps optimization. And development focus on improving the yield and product recovery through the process seem to be pretty standard, but it may pose a lot of challenges as well. Last but not least, and this is more applicable for both cell and vector-based therapies, is that currently we have many new innovative technologies, but those technologies are very expensive, which leads to pretty expensive product development. And with that, I would like to thank the team at Novartis. You see most of the people on the picture, and I'm happy to take any questions.
Thank you very much, Magdalena. I've been looking.
Can I quickly interrupt, Karin?
Yes, sure.
This is the last session, and you can extend it up to 30 minutes if you wish.
I greatly appreciate that. If it's okay for the people, then this would be what we have to do, because as you see, the topics are quite complex and there's a lot to talk about, and 10 minutes is very, very short. So I very much appreciate it. Magdalena, thank you very much for an excellent talk. You also showed us the complexities of the cell and gene therapies. It's not just one therapy, it's multiple. It's the cell therapies and the gene therapies, and then we also have the cell and gene therapies. Thank you so much. There is no questions at this moment. I've been looking to the questions for that. There's also other sessions that will tomorrow that will also talk about cell and genes. Thank you. And now we move on with Dr. Benjamin Rengstel and RNA vaccines. Dr. Rengstel, please.
Yeah, thank you very much. Good evening to everyone, and many thanks to the organizers to make this summit happen also during this pandemic times. So we have heard on RNA-encoded antibodies and also on RNA vaccines and the manufacturing of it from CureVac and Mrna talks, so from Patrick and Don, thanks to them. So luckily at BioNTech, we also have a subsidiary focusing on engineered cell therapies. So I'm in a pleasant situation to present to you our fully owned CAR-T program that we co-developed with an RNA vaccine that drives expansion and efficacy of CAR-T cells after transplantation in vivo. So let me shortly introduce to you the CAR technology. So I think everybody is aware of that the CAR consists of an antibody-derived extracellular domain, so the single-chain Fv fragment, and a TCR, so T-cell receptor-derived intracellular domain that signals like the TCR with the CD3 setter, but also contains co-stimulatory domains to have a full activation of CAR T cells when they encounter the antigen. For generation of CAR T cells, you need a GMP facility to produce the virus supernatant and to mediate the transduction process. And then afterwards, you have a mostly frozen CAR T product that you can ship back to the patient where you received the T cells initially from. There are two CAR T products on the market that have approval already. It was mentioned in the last talk that Mrna has approval, and also Yescarta from Gilead. Both are directed against CD19, so are meant for liquid tumors. And the reason why there is no CAR T product on the market for solid tumors is most probably the problem with the target, because there are no safe targets. So CAR T cells are very potent drugs, living drugs, and you have to make sure that they do not have any on target of tumor toxicities, for example. This brings me to Claudin-6. We investigated this tetraspanin molecule that belongs to a very big family of proteins for decades now, and it has two extracellular domains that are druggable, and we developed a high affinity antibody against it. And the special thing about Claudin-6 is that it gets silenced during organogenesis, as you can see here for lung and kidney as an example. And this makes it as a perfect target for CAR therapy because it is not expressed in any healthy adult tissue. On the other side, as you can see by immunohistochemistry, or here for ovarian cancer as an example, it is highly expressed in a lot of high medical need cancers. And furthermore, expression correlates with disease progression. This is why we believe in that Claudin-6 is one of the only known cancer stem cell markers. We developed a second generation CAR against Claudin-6 with the anti-Claudin-6 single chain FV and a 4-1BB costimulatory domain that shows enormous sensitivity towards its target. So even hard to detect amounts of Claudin-6 mediated a high percentage of target cell lysis. And it's highly specific, so it does not cross-recognize highly related family members like other Claudins. And we have performed tumor spheroid killing assay in a repetitive fashion, meaning this is kind of a stress test. And there we observed that only the 4-1BB containing CARs were able to perform repetitive clearance of Claudin-6 positive tumor spheroids. This is why we decided from a broad variety of scaffolds that we have tested to go with this second generation CAR. Indeed, the stress test was very predictive for the in vivo performance of our Claudin-6 CAR. And as you can see here, very large tumors. This is a xenograft model with some cutaneously growing tumors from an ovarian carcinoma cell line that endogenously expresses Claudin-6. Very large tumors, so larger than what you usually see in the literature, got rejected completely to 100%. Yeah, but we are aware of that the landscape of clinical response in the CAR-T field doesn't look very promising for solid tumors. While you have very good responses in the liquid tumors field, so especially with the CD19 CARs, you lack objective response rates in the case of solid tumors. So here are multiple targets that were addressed. You have to exclude the GD2 because there the T cells get transplanted intracranially, and you don't see a lot of good responses. And we think this is due to the fact that in case of liquid tumors, where you have circulating tumor cells and also healthy B cells that do express the target, you have stimulation of the CAR T cells when you inject them into the circulation. In case of tumors, solid tumors, you have the target within the tumor and there's no circulating antigen. And therefore, if the CAR T cells get transplanted, they stuff in the periphery. And this leads, different than for the CD19 CAR T cells, for example, to a rapid decline that was also shown by Steve before. And the CAR T cells just starve and die off within the first 2 weeks after transplantation. So we raised the question, how can we improve the engraftment and persistence of CAR T cells now that we have such a potent anti-Claudin-6 CAR? And actually our hypothesis is that we want to provide somehow peripheral antigen to stimulate the CAR T cells, and in the best case scenario also to expand the CAR T cells above therapeutic levels in vivo. And actually we have a very potent technology for this already in hand. So we have the RNA lipoplexes that got published in 2016, and they are based on a proprietary formulation. So basically you have in vitro transcribed RNA encoding for T cell antigens. You have our formulation of liposomes. If you combine it, they form RNA lipoplexes. And if you inject those intravenously, so systemically administered, they exclusively go into the lymphatics, so spleen and lymph nodes, and they get uptaken by antigen-presenting cells. So actually by the right cells that you want to address if you want to stimulate CAR T cells. The RNA lipoplex technology is already translated into numerous clinical trials. The recently published data on BNT111, where we used the lipoplex technology and administered it into melanoma patients, showed that the vaccine, first off, is well tolerated, and it shows also clinical proof of concept that those lipoplexes target the lymphoid compartment. And as you can see here, for example, after vaccination in the spleen, there pops up some signal showing T-cell proliferation and higher metabolism. And it could also be shown that in this shared antigen approach that vaccination mounts endogenous T-cell response against the desired antigen. We just exchanged the antigen cassettes against the full-length Claudin-6 because the CAR T cells recognize not MHC-loaded peptides, they recognized the natively folded protein on the surface. And we rebranded it to CureVac. So this means CAR T-cell amplifying RNA vaccine. As a proof of concept from our preclinical studies, we used luciferase labeled CAR T cells, and what you can see here are a lot of mice that show a baseline signal of luciferase when you have transplanted them with 1 million CAR T cells. And then we applied titrated amounts of the RNA vaccine encoding for Claudin-6, and what you can see is a tremendous increase of the signal, meaning that we expand the CAR T cells in vivo very rapidly and very sufficiently, or with high potency. And what you also see after expansion phase, you see a contraction phase. And this reminds of the natural stimulation of T cells and the natural behavior. So we raised the question, what happens if we apply CureVac for multiple times? And as you can see here in the graph, we did the same experiment, but we applied the vaccine multiple times. So we were able to keep the cells at a certain high frequency. Then we decided to go for a treatment break for 4 weeks. And interestingly, the CAR T frequency never dropped to baseline. So gray is the same amount of CAR T cells with control vaccination. And we were able to expand after 4 weeks again to a certain amount. We performed it another time and we saw that we could multiple times expand the CAR T cells in vivo. And then we unfortunately were at the end of the timeline that we were allowed to perform in vivo studies. And here you can clearly see that we also with different detection methods that we enrich for CAR T cells, so here by flow cytometry. And most importantly, we do not only enrich for CAR T cells, they acquire a memory phenotype. So this means that we really mediate persistence to the CAR T cells, not only expanding them. So next, we wanted to know how low can we go and expand the CAR T cells. So what you can see here are the baseline levels if you transplant 10 million CAR T cells, 1 million, 0.1, and only 1,000 CAR T cells. When we applied the vaccine twice in this case, in a weekly fashion, we expanded by multiple logs to therapeutic levels. And here, regarding therapeutic levels, you can see here that we did a titration of the flat doses of CAR T cells, so 10 million, 100,000, 10,000 CAR T cells per animal in our xenograft tumor model. And we could clearly show that 1 million is the needed dose to reject tumors completely. Then we decided to use the insufficient dosing of 100,000 CAR T cells and to combine it with CureVac. And after 3 vaccinations, we saw tumor shrinkage, speaking for a restored anti-tumor response of the CAR T cells. We also performed the same experiment in immunocompetent mice, very important. So these tumors are harder to treat than in the immunodeficient models where you use a xenograft and human CAR T cells. Here we have a fully syngeneic model, and interestingly, one vaccination alone was sufficient to boost the insufficient CAR T dose to reject the tumors completely and forever. And this is due to the missing mismatch between the antigen presenting cells and the CAR T cells, because this is a fully murine system. And this is what we also expect for the patients later. So then, while publishing the paper, to be honest, the reviewer asked the question, is this only a numbers game? And we had to come up with a smart experiment. So what we did is we used a low dose, expanded this dose to the same amount of a high dose non-expanded CAR T cell. And we extracted the CAR T cells from the mice at certain time points during expansion phase, during contraction phase, and so forth. And what we observed is, in every analysis that we applied, and there's way more that we did, that we see a favorable cytotoxic profile of the expanded CAR T cells. So they do better secrete, they do better lyse, and so on. So they gain a better polyfunctionality. This brings me to the summary. So I introduced to you our CAR T program called BNT211, where we fully own, manufacture Claudin-6 directed CAR T cells in our own GMP facility. I explained to you our CureVac approach, that we have the RNA lipoplexes to deliver CAR antigen, and that we want to combine both for in vivo expansion of CAR T cells. And our aim is to keep the CAR T cells in a certain therapeutic window. And this brings me to the pleasant situation that I can introduce to you or announce to you that we have opened a clinical trial to evaluate the safety and preliminary efficacy of Claudin-6 CAR T cells with and without our Claudin-6 vaccine. And this trial is now live, and it will be conducted in Germany at 6 centers and also in the Netherlands at 2 centers. And we are really looking forward to the performance of our Claudin-6 CAR T cells, and are hoping for very nice responses, different that normally observed in the CAR T field treating solid tumors. And as a last point, I just wanted to say that CureVac has a universal character, so we have many more targets in the pipeline, and we have tested also CureVac with established CAR T cells from different groups and different companies, and we can say that it works basically in every case. And yeah, this is, for example, Claudin 18.2. It's also a Claudin, a tetraspanin. We have a nice antibody against it. We made a nice CAR against it. And we already have the data that this is of high potential for a lot of patients. In this case, patients with gastric cancer or pancreatic cancer, lung cancer. And I have forgotten to mention that Claudin-6 will be a basket trial for gynecological cancers, testicular cancers, lung cancer, gastric cancer, and many, many more, all that are Claudin-6 positive. And with this, I would like to thank the whole team, because this was a real team effort, all the partners involved, of course, Ugo and Özlem for kicking off such fancy programs, and all of you for your attention. Thank you.
Thank you very much, Benjamin. It looks like the future is here already.
Thank you.
It's nice to see the CAR T field move to solid tumors. This is quite exciting. Do you expect with your so it's going to be a transfusion, I'm guessing. Do you expect also these side effects that we have for the liquid tumors, as you call them, like the CRS, for example?
No,
or do you think it's
honest,
going to be nicer?
Not with the monotherapy, because Claudin-6 is, a target not present anywhere in the body, only located to the tumor, and the CAR T cells have first to enter the tumor. So I think, and then they have to eat through multiple layers of cancer cells. So I do not expect a full-blown reaction in the beginning, no CRS, maybe no tumor lysis syndrome, but I'm not sure about the vaccine. If it's really as potent as we have observed in our preclinical studies, there's a chance for transient CRS-like symptoms. Yeah. So we will monitor this.
I've been renewing my questions, but there's not much. So hopefully we have some questions offline for you. But this was very exciting. All the best to BioNTech. And we'll go on then with the next one, which is, and thank you also for keeping the time, with Dr. Stefan Halbherr, who's making medicines with new behaviors.
Good evening, or good morning, or good night to everybody around the world. It's my pleasure to present to you our latest understandings in how making medicines behave in different ways that usually they are not able to do in the in vivo setting. So to give you the reason of our inspiration, we started in 2013 when the Nobel Prize was given to folks that discovered how vesicles actually traffic inside of cells, so they get modified throughout the Golgi, and that has been a very interesting story. And most likely transporting systems are very sophisticated and ubiquitously used in the body. And I think one of the largest questions to date are how can we make medicines behave in the way the logic of the body is basically requiring. So we went on to this mission in making medicines this way more effectively and effective and less costly. And I wanted to show you actually a story of our own research, how we went from making liposomal doxorubicin, our lead product called Talidox, into a 100% biological entity that is called talinurine. It's a liposomal GM1 ganglioside. And there will be a talk about this product by Camille Peitsch tomorrow at 15:30. There's a lot to tell about both products, and I actually won't tell too much about each product's peculiarities, but rather tell the story of how we discovered the different aspects of them and what drove the decision making. So essentially, we tried we tried things that are not supposed to work. Whenever somebody says that's probably not going to work, it's also a good indication that if you try really hard, sooner or later you might get it to work, or you find something really interesting. And learning from these kind of processes, of course. So putting these two products left to right, I think we have this sort of anthropocentric approach where we try to rationalize everything into, particle size. Obviously, nanomedicine plays a big role. There's a lot of knowledge in there, as well as a composition of these kind of medicines, including the composition, how they are made, either cationic, anionic, what kind of surface-specific attributes they have, and how the targeting of the medicine is actually made, either by pegylation of the surface or attaching ligands or using magnetic fields and many different strategies. So when we look into how is nature actually doing it, I find nature a great source of inspiration. I find that particle size may not be that big of an issue. Somehow biological structures are really large. If you look at neurons, for example, they can be up to 2 meters in length. On the other hand, intracellular vesicles are very small, or viruses are around 30 nanometers. So essentially, I think from large to small, everything can work. And what we really need to look at is the complexity of the system and how it's responding dynamically with the membranes that have static charges or ion channels to react to different fields. And I think there is a lot of interesting research going on in that area. So we can, when we compare the two, we see that, of course, there are some body foreign substances which are foreign to the logic of the human body, which I think is an important aspect. So for liposomal doxorubicin, body foreign is, of course, doxorubicin as well as the surface, which is the polyethylene glycol 2000. Whereas on the other hand, we have 100% endogenous substances, which comprise sphingomyelin, cholesterol, and GM1 ganglioside. So fairly simple up to that point. And how did we actually discover this green particle, and what makes it so special is that when you load this particle with doxorubicin, and we were loading all kind of different particles with doxorubicin, we found a very surprising effect, which was that this drug, which is normally a very well-tolerated dose of doxorubicin, caused actually death within 24 to 48 hours after injection in mice. So that was a very big surprise to us. I think it is, of course, an unfortunate finding on one hand. However, on the other hand, I think we were the first who sharpened doxorubicin in a way that it can actually kill the entire organism within one single injection, which typically it doesn't. So we knew that we definitely changed the biodistribution of the drug in a way that it somehow seems to hit to vital organs which are very sensitive to this drug. So I think it might have been the heart or the brain that would react in this way, and we of course followed it up. So I think it's somewhat a happy accident, and we started to identify each parameter that has an influence on the particle behavior of this green particle, tallynurine, with doxorubicin. So first of all, we thought, we must have made a mistake, and we threw away all the reagents we had that we thought, might have some level of contamination whatsoever. We did the same again, and it was very reproducible. Then we tried to do what typical in liposomes is done very often. We pegylated the surface. And interestingly enough, we saw a delay of the effect. So mice started to lose a lot of body weight and also died after 2 to 3 days, unfortunately. And we were always trying to target this to tumors, of course. And we were really puzzled in why is this happening? So we thought, okay, let's leave out the doxorubicin. That should certainly be no problem at all. So we left out the doxorubicin, and we saw that the nanomedicine itself is, as expected, perfectly well tolerated, and there's body weight gain were totally similar to controls. We also left out GM1 ganglioside because there is the notion that these surface-exposed molecules could generate some level of targeting. And of course, there are many lectins or other proteins that bind to GM1 carbohydrates, and there might be the hypothesis that GM1 is generating the targeting. And interestingly, it isn't. So in this case, we found that biodistribution was totally dependent on actually the sphingomyelin and cholesterol, as well as the particle size, most likely. And I'm sorry for the orientation of this picture, but the chemical software wasn't able to depict it differently. I think it was very interesting to see that actually the difference in behavior, what we found is that it's because of the linkage of the fatty acids of the phospholipids that actually make up this very big difference in how doxorubicin affects the body, which I thought was really surprising. I would have rather expected that, you know, charge carrying molecules such as the phosphocholine group would generate a large portion of the effect, but it somehow doesn't. So it's rather also that the underlying bonds of the fatty acid chains that make up a lot of biodistribution of liposomes. And this is also in line with our findings of how bacterial toxins, molecules that have withstand many years of evolution, they recognize also, interestingly, the saturation of fatty acids. They recognize the saturation of fatty acid chains in the body, in the liposome membranes. So also hinting that most likely there seems to be biologically important mechanisms that recognize how the fatty acid chains actually are saturated and packed into a membrane. And this concept is, of course, interesting to scavenge bacterial toxins and neutralize bacterial infections, which is one of the research areas we are investigating. So, short summary, I think surface is not the only part of the puzzle that really generates biodistribution of particles. It seems to be more complicated. And interestingly enough, when we leave out the doxorubicin, we end up with a very neuroregenerative medicine, and there will be a talk about this, investigating each single detail, how we can use it in Parkinson's disease and ALS. And just to highlight one important aspect of it is bioelectricity. And it's a new way to recharge basically the brain, because it has been shown that the brain and the glia carry actually the most net negative charge of all the tissues in the body. So Vmem potential of -70. And there has been many studies that show when the brain develops, it basically from embryos, the charge and the ganglioside structure becomes more and more sophisticated, and as the brain ages, it sort of loses that multitude in complexity and starts to degenerate. So this medicine basically brings back quite some molecular complexity to the brain that is somehow struggling in keeping up its complexity. So also interesting notion that cancer is somehow a over-regenerating process, and it carries a net negative overcharge compared to healthy tissues. So I think that points towards new treatment modalities to use charge-carrying molecules, such as GM1, for example. I think it's very important to really pick the molecules that carry these charges and to use them in a therapeutic setting. But I think given that there is a lot of potential in neurodegeneration or cancer or many other diseases, since charge or membrane charge itself is involved in very fundamental processes such as organogenesis, morphogenesis, and cancer. So I know this sounds a little crazy, and I wanted to show at least one picture before the talk of Camille Peitsch to really get you interested in the topic. So this is a section of the brain called substantia nigra, and we injected red-colored talinurine liposomes, and we sliced the brains and looked at them in confocal microscopy 24 hours post-injection. And you can really see that some cells have taken up a lot of red dots, where you see, for example, the zoom on the right, whereas some other cells really don't. And I cannot really tell yet why some cells take up a lot of the particles and others don't. But interestingly enough, this nanomedicine is very simple in design, and apparently reaches the brain to a very high extent, and can exhibit neuroregenerative effects. So far to neurology, and I wanted to quickly draw your attention to the oncology, which is similarly important. So there has been, of course, the never-ending question how to get intravenously injected drugs accumulating into tumors, and I'll elaborate on that on my talk also in higher detail on tomorrow morning. So I just wanted to have an introductory comment here that, doxorubicin has been invented in 1950, and 45 years later, the first liposomal formulation, well known as a calyx, was registered, and 25 years later now, there is still no follow-up, basically, which I think is unfortunate, and I think we somehow should find ways to have a more higher frequency of how follow-ons of successful medicines really enter the market. So I think it's key to keep it simple, to have concepts that are actually translatable easily, scalable, fast, and cost-effective, and to really also take what has been working empirically and build an advantage edge on this. And also in terms of regulatory pathways, I think one should maybe look at new ways how to get drugs into markets, and looking at the benefit risk cost ratios. So in tumor targeting, I think it's essential that the particles are really small, and Talidox is 35 nanometers. It's the world's smallest doxorubicin, and it's very high in particle numbers. It's 9.3 times 10 to the 13 particles per milliliter. And that has some serious advantages when it comes to performance in vivo. So there is an apparent tumor accumulation effect, and uptake by cells is of course faster when particles are smaller, as well as the diffusion through dense extracellular matrices is faster when molecules or aggregates are smaller. So there's a couple of advantages, and there is a faster drug release if you don't put too much drug into each liposome, which I want to discuss with the recently published work. So there has been a meta-analysis along with some original data showing that the higher the particle number of the nanoformulation, the more drug actually enters the tumor, whereas liver clearance goes the other way around. The higher the particle number, the lower the liver clearance. So I think we might have been running a little bit in the wrong direction in trying to maximize drug loading into particles, and probably the other way around is might be achievable, more desirable, that we try to minimize actually the drug loading. And while minimizing drug loading to particles, of course, the toxicity of the nanomaterial itself will be key in actually giving large amounts of the nanoparticle and small amounts of the drug per particle.
Well, I'm sorry, but we're getting really out of time. Can you wrap it up?
Wrap it up, yes.
Thank
so I
you.
think medicines should be as natural as possible, following the logic of the host system, so the human body. Science matters, but not really in everything. Nano-organization is probably key, and the medical entity itself can be large or small. There is a lot of potential in bioelectricity for regeneration and cancer treatments and more. Tumor targeting is, I think, desirable and quite a challenge, but it's feasible. And of course, medicine should be affordable to all. So simplicity in the design of the nanosystem is key. And I think for managing innovation on the long run, we have to master manufacturing appropriately. That would be key. Thank you very much.
Thank you so much. Out of the box thinking is always something that I appreciate, I have to say personally. And we have two more speakers. I hope you guys are still on it. I would like to move on with Dr. Dan Pear, who is also going to talk about manipulating cells' function with novel lipid nanoparticles, and looking forward to this one. Dan Pear, please.
Yeah, thanks. But I can't share my screen until...
Stefan, you have to get out.
Okay, good. No worries. Okay, so it's a pleasure, unfortunately, to be here in this such an event. But I want to remind Beat that there are other days, for example, this one in 2017. And I think that was a very nice gesture. And I hope that next time we will all be able to meet again. So my lab is studying how to manipulate cellular function in order to generate novel therapeutic approach to treat mainly leukocytes implicated disease. It's a huge field that includes blood cancer, inflammation, and viral infection. We are very occupied in all those bluish diseases. And I don't have time to talk about everything, but we are one store shop, I would say, or one store that has everything from delivery system. We generate lipids, we generate monoclonal antibodies, protein engineering, and we work with nucleic acid. We do drug discovery, and hopefully want to combine all of them. I'm not going to talk about what we have done, but very briefly what we are doing now. So we are intrigued from the generation of lipid nanoparticles over the years, ionizable lipids, and we decided that it's a good time to really look carefully at those structures, for example, the D-lim MC3 DMA. So based on this gold standard, we have synthesized 60 lipid families and counted to about 2,000 different lipids by changing the cationic head groups, the linkers, the hydrophobic tails, and basically show that we can very quickly test them, not all of them, unfortunately, but some of them. And so we generated a lot of knowledge about what can they do, how toxic they are, how good they are, and for example, since we are interested in immune system, how immune toxicity they have. Or in this example, how they have really minimal liver toxicity. I won't say zero, but I say minimal in non-human primates. And so over the years, we are using microfluidic systems to entrap nucleic acid from siRNA to mRNA and other nucleic acid type to make monoclonal antibodies. And then we were using reduced monoclonal antibodies in order to conjugate them into the surface of lipid nanoparticle. This is a very inefficient way, and probably because it's not an industrial way. And in fact, you cannot get this very nice picture here, so you cannot really control the orientation. So therefore, we decided, to move from chemical conjugation that has lots of issues. The efficiency is very low. Antibodies are not alike. Some of them have glycosylation. And of course, you can have a poor binding because you have Fc exposure, and these will be taken out dramatically quickly by cells that have Fc receptors. And so from chemical conjugation, we move to what we call a linker universal strategy of affinity, going back to biology. And we generated basically an antibody that recognized the Fc portion of an antibody. Therefore, you have the same Fc, you only need one chemical conjugation, and then you can actually control the orientation. We've published numerous papers over the past years. I'm going very quickly about this because I don't have time, okay? But as you understand, orientation is a key. If you can control the orientation, you will be very, very effective. And we can go as low as 250-fold less antibodies very, very effectively. And so behind this idea were two former PhD students of mine, Ranit Kedmi, who is now an immunologist at NYU, and Nofar Vaiga, who is now in Leuven, a cell biologist. And basically, we came up with this very elegant approach that gives us the opportunity to manipulate leukocytes in vivo in subsets of cells. And so Our collaborator is Itai Benar, is a protein engineer, and Mark Belke from IDT for many, many years. And what we have done, we started with a complete IgG that we generated. Then we made single chain, and in one transcript, we found a sequence that can lipidate the single chain. Therefore, these guys can now be incorporated into the surface of lipid nanoparticles, and then you can go to different directions. So this was published about 2 years ago. From there, we moved on and published numerous papers in this approach, targeting different subsets of cells, immune cells, and recognize also new therapeutic targets, mostly in IBD and in blood cancers. So again, this is the versatile approach. If you put, for example, Cy5 siRNA, and you put on the surface either an anti-integrin beta-7, anti-CD3, anti-CD4, anti-CD25, you inject them IV. In 1 hour later, you can isolate subsets of cells, and you will find out that the anti-beta-7 will go to all leukocyte subsets, the anti-CD3 only to CD3 cells, CD4 only to CD4, and CD25 to a subset. We also showed that it's therapeutically relevant to do this in one model of colitis and one model of mantle cell lymphoma. And again, here we use as a target cells an anti-Ly6C high-expressing cells, basically inflammatory leukocytes, and a target gene of TNF. Okay, and this was blinded, it was done in Charles River, and you can see that the data supports this. If not, you can read it. And also for mantle cell lymphoma, we use receptor targets as beta-1 integrin, CD29, and porolykinase 1 as the proof of concept gene. And we also developed a model for mantle cell lymphoma a few years ago, it was published in PNAS, and then we used this model basically to show that it's therapeutically relevant. We also were very intrigued about the ability to produce proteins. And so therapeutic protein expression, as you know, for vaccines, RNA vaccines are now very popular, cancer therapy, protein replacement, and immunomodulatory. Actually, we are the first to show systemic cell-specific delivery of mRNA, and basically using exactly the same approach in 2018. Again, in a colitis model, this was the first paper, then came other papers, but basically here we show that we can target inflammatory leukocytes very selectively, very efficiently, and you can see that they're also expressing in the intestine, in the spleen, and in the liver. These are the Ly6C high population, and it has also a therapeutic approach, but basically we chose as a proof of concept the expression of IL-10. We inject them systemically. The idea was that maybe they will target cells that are nearby cells and will enter the cells, produce the mRNA, and then the mRNA will be released, the protein will be released and will change the what we call gut microenvironment. And in fact, this was was happening. And in all the parameter, it looks very, very nice. And it also looks as a safety profile that this strategy works well. From 2018 until now, we published numerous papers with this approach. And now I'm going to new data that I haven't showed before, which is a paper that will come soon on CRISPR-Cas9. And basically here we decided to use our ability to target specifically mRNAs, and using Cas9, we also do Cas12 and 13, as well as guide RNA. And so we decided to do this in cancer. And in this approach, basically it's exactly the same approach. We put on the surface, in this case, we can put an antibody against EGFR or any antibody to any target that we want. We use 2 animal models. I'll explain. One is the Maureen 005 GBM model that was developed by Inder Verma and kindly gave to us a few years ago from the Salk Institute. This is an orthotopic, syngeneic, super aggressive, highly infiltrated mouse glioma. And we also use the human OVCAR8 xenografted disseminated model. Here I just want to show you that you basically get very, very efficient gene cuts, basically on the indolent next generation sequencing, 98%, which is very, very high in vitro. And I'll show you also in vivo. And basically this was a proof of concept. So you can see that guide RNA for PLK1 compared to a control guide RNA for GFP, you get a dramatic shift here. And also the cell cycle here, you see dramatic shift. And in the animal model, basically, we stereotactically injected basically cells that express GFP, and then about 50 microgram per kilogram of Cy5.5 label CRISPR-based LNPs. And you can see that they actually disseminated very nicely inside the tumor. I don't know if you can see this, but we can see. Then we repeated this also to look at GFP disruption. This is eGFP disruption. We also saw this. And then we saw basically that staining with cleaved caspase-3 as a measure for induction of apoptosis, also very, very specifically only in mice treated with this guide PLK1 and not with the GFP1. So you can see here the caspase staining. Hope you can see. Okay. Next, we implanted 005 GFP M-cherry luciferase cells stereotactically into the hippocampus. And then we can do 7 days later an imaging with an IVIS, then inject LNPs directly into the tumor bed. And this is only a single injection. So either a PBS, a single guide GFP, or a single guide PLK, of course, with the CRISPR one. And then you can monitor the growth of the tumors. And this is what we got. Each group is 15 mice. Okay, so it looks very, very convincing. And if you look at the increased median survival is by 50%, the overall survival change by 30%. And if we look at in vivo editing by next generation sequencing is 70%. All our NGS studies were done in collaboration with Mark Belke and IDT team in Iowa. And so here we have 30 mice per group. So these are very, very big groups. And a lot of work for the, of course, for the postdocs and graduate students. Then another one, I want to show you a disseminated model, which is the targeted one. It's basically an ovarian OVCAR8 disseminated model. And again, the same thing happened here. PLK1, you get almost 100%, 98% gene disruption. If you see the control, the GFP locus, it's basically here. So it's very specific. And then again, you can see here the therapeutic genome editing that you can see in cells and in vivo. What we have done here, it wasn't a single dose, but 2 doses. And we follow up for 60 days. So the glioma was 1 dose and follow up for 60 days. And here we have 2 doses and follow up for 60 days. This is a disseminated IP injection of the cells. And what you can see here is actually a dramatic change in the survival here, as well as the tumor development. And again, assuming that those mice will develop tumor, we know that they are developing already in week 2 and 3. So you'll see that there is a growth here in the tumors, 10 mice per group here. And again, 10 mice per group. Here, the overall survival is 80%. In vivo genome editing by NGS is 80%. And this is the highest in vivo genome editing that was published until now. So I hope that this is good. We also did safety profile. And then at the end, I just want to quickly say that where are we standing? Well, lots of things we can talk about. For example, the kinetics of this, fast internalization, burst release, et cetera, et cetera. But I think we have very, very interesting lipids, as well as a targeting approach that could be used for drug discovery, in vivo drug discovery, as well as for potentially therapeutics. And just the end, the people that worked with us here on this project, and the funding agency that over the years help us. We have a collaboration very active also with BioNTech and ART, as well as previously with Alnylam. And we are funded by the Hemsley, NIH, and ERC. And that at the end, I just want to show you a short movie, which is actually a fun one.
In inflammatory disorders, the sick cells in our body are our own white blood cells. Only instead of defending the body, they attack it. The best way to fight the complexity of human diseases is to deliver the therapeutic payload directly to the sick cells. A promising technology for combating diseases uses special lipid nanoparticles. These nanoparticles contain RNA molecules that can change the way cells produce proteins and can modulate cells' function. The nanoparticles are supposed to deliver the RNA directly to the sick cells. The problem with today's technology is that the nanoparticles hardly reach immune cells, but rather affect other healthy cells instead. We at Pear Lab have developed a new platform called Asset that can send RNA nanoparticles selectively to the correct cells. While existing methods of connecting nanoparticles to antibodies are extremely complicated, Pear Lab's method is a matter of simply mixing two solutions. And the nanoparticles are decorated with an asset linker, which then binds the antibodies. Each type of antibody knows to attach to a very specific type of cell. So when they decorate the nanoparticle, they will direct it only to the targeted cells. This method means releasing the RNA right where we want it, affecting only the right cells and saving the body from harmful side effects. Asset nanoparticles are a novel strategy to fight against inflammatory diseases and even cancer. And this time, it's personalized.
Okay, that's it.
That was a very cool movie. Thank you. I also think of allergies, which is also doing crazy stuff with our bodies. So I hope you will find something that you will reduce the amount of eosinophils in my circulation in spring next year. That would be great. Less sneezing, less coughing, less asthma. Perfect. Thank you very much, Dan Per. This was a very inspiring talk, and it was a lot of fun to watch the last movie. So let's go to Professor Dr. Maria Jose Alonso and then conclude the session. Thank you, Dan. It was cool. Thanks.
Here we are. So hello to everyone from Santiago de Compostela in Spain. This is my research institute. And today I would like to tell you about how we design nanocarriers for the delivery of proteins and RNA molecules, something that we have been doing for 3 decades now. So the profile of my lab is biopharmaceutical. I mean, we work at the School of Pharmacy as well. And therefore, my approach towards the use of nanotechnology has this pharmaceutical component. So we really like working according to a rational design, which takes into account, obviously, the nature of the drug to be loaded and the barriers to overcome. So it is certainly true that people publish lots of papers about how to deliver a molecule inside cells with in vitro studies. Obviously, this means not much. We do care a lot about all barriers that drugs need to overcome before reaching their target. And obviously, there are many barriers, as we have been listening to several people here today. So we designed these nanocarriers taking into account the drugs, so obviously it's different, an RNA molecule versus a peptide, and the kind of barriers, which also depend on the modality of administration. So because of our translational view, we also like using biomaterials that are recognized for their safety record, such as polysaccharides, polypeptides, surfactants, oils, and very classical excipients. So, there is something going on. I cannot move to the next slide for some reasons. Okay, here it works. Okay, so for the sake of short time, we have today being the last one. I don't really want you to get too tired, so I'm going to mention only a few applications that we are dealing with nowadays in my lab. Obviously, nanotechnology does not have any limitation in terms of the kind of therapies that can benefit from it. But today, I like to concentrate on vaccination, especially by the nasal route. A field that we have been covering for 3 decades now. More recently, we also started using the brain for delivering polynucleotides to the nose for delivering polynucleotides to the brain. And obviously, as many people working on nanomedicine, we are trying to do our best using our technologies for treating cancer, and in particular for reaching undruggable targets that have not been reached so far with small molecules or with large molecules. So in that case, we are using monoclonal antibodies and RNA. So let's start by antigens, by vaccines. So as I said, 3 decades of work using biodegradable nanoparticles for the delivery of these proteins. Actually, since I did my postdoc with Bob Langer, at that time we were with microspheres, then we moved to nanoparticles. And you may wonder what has changed over the years. So many things have changed over the years, obviously from the nanotechnology side, but also, and very importantly, from the immunological side. And so we have been working with all kinds of antigens, going from classical proteins, peptides. More recently, we have been concentrating on HIV vaccine, 2 different vaccines actually, based on peptides, based on mRNA. And obviously our work on vaccines has led us to a COVID vaccine in which we are also involved, and I will tell you a few words. So what has changed? Many things have changed, the kind of biomaterials, the localization of the antigens within the nanomaterials, the incorporation of immunomodulators, everything has changed, and we have much more information, obviously, nowadays than we had 3 decades ago. So one of the HIV prototypes we have been working with is this illustrated here. I mean, the majority or all the vaccines, as far as I know, going to clinical trials for HIV have been mainly based on proteins. However, these guys from Manitoba, who we met at the Gates Foundation, had the idea of using small peptides rather than proteins, targeting specific epitopes. So they told us, we have 12 peptides and we would like them to arrive to the same dendritic cell population. And therefore, we encapsulated these 12 peptides with the same biomaterials, different lots. We made a cocktail of them and we administered them to mice and then to macaques. It has been 10 years of work. We have even done the scaling up of this prototype thanks to the financial support from the European Commission and from the NIH Institute. We have done the in vivo studies, as I said, in several animal models, but the most relevant probably are those indicated here, the macaques. We have done this series of experiments 4 times already, and I'm going to the point. We are talking so much nowadays about IgG responses, T-cell responses. At the end of the day, what we want to see is that by challenging the animals, by challenging people with the virus, they don't get infected. So we have many data published until now, but this is probably the most important graph. And this means that after 8 challenges, 80% of the macaques remained alive, non-infected. That is the key point. And this was following intranasal administration of our nanoparticles containing 12 peptides. Now we are trying to reduce the number of peptides. Things are not going that well. I mean, it seems that we require a minimum number of peptides, but we are looking forward going into the clinical development phase at this point. So, as I say, this took us to the COVID vaccine. Of course, we built the Covarna consortium mainly with people from Spain, but also from Belgium. And in this case, our way has been, as you know, several companies presenting here today using RNA. So we are using two kinds of RNA, two different molecules, one intended to produce a T cell response, another intended to produce a B cell response. So we are co-formulating these RNA molecules in our nanoparticles and obviously administering in vitro, in vivo, and then now we are looking at the IgG responses, T cell responses. I cannot tell you much. We are now in the middle of the in vivo experiments. Things look apparently well, but I don't want to say more because this is, you never know. Okay, so what have we done in this big consortium? Obviously, there were lots of people, trying to design the RNA molecules, trying to do with artificial intelligence and a number of immunologists. Our work has been to design the nanoparticles. And we have relied on everything we found in the literature, our decades of work in the field, and we have selected 150 prototypes we have tested already. We had a kind of target product profile, although this is not like the classical one you see, but we fixed a number of properties that we wanted for this, and we selected 75 prototypes that were tested in vitro, and then 10 of them have been tested in vivo using obviously different kinds of RNA, but eventually with the two RNAs for COVID vaccine. So this is where we are now. And as I said, based on our experience on nose, on delivering drugs to the nose, we decided to explore the possibility to deliver RNA from the nose to the brain. And well, the barriers are kind of very limiting. However, it is also true that the BBB is also very limiting. So we decided to explore this way, and we built some prototypes that are illustrated here. In this case is for mRNA, microRNA 132. So we made this kind of nano complexes of RNA with different condensing agents, and we protect this composition. This is not PEG as usual, but with different polymers. So we selected some prototypes with these specific properties. It's important that I do believe particle size, somebody was talking in media about not being important. I do believe particle size matters. Obviously, it's not all. The composition is also very critical. But so it is important that the particles are stable in biorelevant media, something that is not very often is not the case. They have to be mucodiffusive. They need to have a very high loading because we really want to use the minimum amount of particles, in the sense that, we don't really want a highly concentrated solution. They protect RNA from degradation, and we could see an enhanced uptake either in vitro or in vivo. Going to the in vitro, I know it's easy. In vivo, going to this graph, you can see that we have a statistical significant difference in the sense that the amount of microRNA that can reach the brain with our nanoparticles is much greater than for the control. So this is nose to brain RNA. So just going to the end in the field of cancer, obviously we have done a lot of work with small molecules, chemotherapy, but then today I'm going to concentrate on monoclonal antibodies because despite the advances, monoclonal antibodies have a number of limitations. And a very important limitation is that all monoclonal antibodies marketed so far are only intended to reach extracellular targets or targets on the membrane of cells. So we are trying to reach intracellular targets, and for that we have been using almost for a decade nanotechnology. So this is a new concept. Dan Pierre was talking about functionalizing particles with antibodies. In our case, the antibody is the oncological molecule, the pharmacological entity. So it's not a targeting ligand, it's a pharmacological entity that we want to deliver inside the cell in order to reach intracellular targets. So this is the kind of formulation we have developed until now. We call them multifunctional polymeric nanocarrier. they have a small size. They are made of regulatory acceptable materials. We have done some scaling up. The technology is extremely simple. We can freeze-dry this composition. And something very important from the biological point of view is that we have found that these nano capsules that you can see here do accumulate quite importantly in different tumors that we have explored until now, and also in the lymphatics, where we have in some kind of cancers, the metastatic spreading. So the feasibility of the technology in terms of monoclonal antibodies delivery has been done already for 6 monoclonals, and we have extensive proof of concept studies for 2 different monoclonal antibodies. Just to show you, the first thing we wanted to do, obviously, and this was already published a while ago, Anna Kadete, who is now at Mrna, did her thesis a while ago in this field, was to prove that the monoclonal antibody could go inside, as you can see in the green dots here, and it was not colocalized with the endosomal compartment. So we did have endosomal escape and additional mechanisms of entry of our antibody inside the cells. So as I said at the beginning, it's not only important to do an in vitro cell culture study and to see that particles go there. Of course, they go there in vitro. What happens in vivo? In vivo, we did find the same. So you see here the tumor with the monoclonal antibody inside the cells, inside the tumor. Obviously, the first thing we had to do before going in vivo was to check the stability in plasma. As you know, many nanocarriers disassemble in plasma. They are not stable. And we could see that we can protect the antibody inside these nanocapsules, and they are stable and they reach the tumor as is shown here. So, okay, so we did our studies initially with anti-Gas-domain monoclonal antibody, which is, not that well known at this point. There is under research, but then we wanted to move to another more relevant target, also because we started a company, Libera Bio, trying to explore this technology for the intracellular delivery of monoclonal antibodies. And then we chose probably the most relevant target and the most challenging target we could think of. And this is the KRAS target, which is highly present in many different kinds of cancer, but in particular in pancreatic cancer, lung cancer, and colorectal cancer. This target is kind of hidden in the inside of the membrane of the cell. So there is a challenge, of course, delivering the monoclonal antibody inside and reaching this target. We haven't done plenty of in vitro studies, but just to go to the point, we do have a series of in vivo studies looking at the effect of this monoclonal antibody, either alone or in combination with other treatments. And we did see when we administered the monoclonal antibody alone, we did see a significant decrease in the tumor growth. When we co-administered with other treatments, we did see the elimination of the tumor growth. Obviously, this was accompanied by extensive necrosis in these tumors. So this is all I wanted to tell you today, because I know everybody's tired and I wanted to be really in time. So just tips to keep in mind, that we keep in mind, we need to tackle multiple barriers, not only cell barriers, and they are very consistent, but nanotechnology can help to overcome these mucosal barriers, skin barriers, many different barriers. It is important to have a kind of target profile for your prototype from the beginning. And up until now, I can say we are in preclinical evaluation of the HIV vaccine prototype. With COVID, we are already, trying to go as fast as possible with the scaling up and everything. But in the case of anti-KRAS, we are also at that level, still preclinical evaluation, but quite advanced. And in the case of nose-to-brain delivery of siRNA, we are collaborating with some companies, but we are still at the early phase of the proof of concept studies. So I should thank all the financial support from many different institutions from Spain, but also from Europe, from the US, and thank you for being until the end, and stay healthy. That's a pity we cannot meet in person as usual. Thank you so much.
Thank you very much, Maria. This was a great session all the way to the end, and I have to say I was not exposed to nanoparticles before. I'm really only working on cell and gene therapies, but this has convinced me, and hopefully we will use that technology in combination with our cell therapies. That would be cool. Thank you so much. I would like to close the session. The discussion should be and debate should be in lounge 1. I'm not really sure what we have to do now to get to the lounge, but I hope that somebody else will tell us.
You can just
go ahead.
You can go in the stream, Cleanam 1, 2, 3, and then you can enter a room. On the bottom, you see stream. Now you see live inbox.
Okay.
You can go.
Perfect.
I would like also to say afterwards something.
Thank you very much to all speakers. Thank you very much to everybody who listened. And I hope that we will see each other in person next time. And I have to second what Maria said. Please stay healthy and be safe. Thank you.
I would like to say that I hope that the 200 people that have now been listening until now to you will at 8:15 tomorrow be fit for going to the late breaking sessions to the 3 platforms that will be offered tomorrow. I wish you well, and I thank you very much that you have been here.